Excuse me. अरे the head. It's okay, bro. Hi, Pushkar. Pushkar, yeah, it is. Now I'll give test, okay? I can't see your texts. You can talk with my friends if they are here. Shubham, Alex, are you there? Nidhi, Sarvajita, Shri, Miban. I think no one is there. I'll give test. Jacket क्यों by the way? I live in Sikkim, bro. या तो मैं तुम्हें अच्छे से समझा सकती हूँ या तो मैं तुम्हें इग्नोर कर सकती हूँ. I choose to ignore you. I thought there is a glitch in Discord, Darwin, or my network is lagging. Which chapter test? इसका नाम क्या था? Sell the unit of life. डाउनलोड नहीं हो रहा है आदर. आई थिंक कि अब तो ऐप में ही ग्लिच है क्योंकि नेटवर्क थोड़ा स्टेबल हो गया है ना. आई थिंक अगर फिर से एक बार ट्राई करूँगी, नहीं हुआ तो मैं छोड़ दूँगी. आई कांट वेस्ट टाइम ओवर दिस चैप्टर और दिस टेस्ट. दूसरा वाला पढ़ने लग जाऊँगा. हाय माँ, आर यू स्टेइंग इन आउटस्कर्ट ऑफ सिक्किम? अय्या, didn't I told you before, Aban? Yeah, you guys can join Discord. डिप्रेशन क्या होता है, स्ट्रेस क्या ही होता है ना. पढ़ाई करते हैं. ये आदर्श ने एक वीडियो अपलोड किया था ना, मुझे तो वही याद आता है. डिस्प्रेशन चल रहा है, डिस्प्रेशन नहीं, डिस्प्लैट चल रहा है, डिस्प्लैट. वही वाला सिचुएशन हो गया है. लेकिन अभी तो ना डिप्रेशन होना और एंजाइटी अटैक आना नॉर्मल है. आई थिंक एग्जाम पास में है ना. Breathing, struck body fluid, biomolecules, animal kingdom, morphology, cockroaches. Cell cycle, cell division. Okay. लेकिन हैंड राइटिंग का मैं क्या ही कर सकती हूँ. I'll study now. ये टेस्ट नहीं होगा, मैं फिर से इसको ना ऐप को डिलीट करके अनइंस्टॉल करके फिर से रीइंस्टॉल करूँगी. लेट्स स्टार्ट. नीट नहीं हुआ तो फिर से दे देंगे, क्या है इसका. नहीं हुआ तो फिर से एक और अटेम्प्ट देंगे. नहीं हुआ तो ये फिर ना होने के बाद देख लेंगे, क्योंकि अभी पढ़ लेते हैं भाई. 13 मिनट है. थर्ड सेशन इसके बाद स्टार्ट होगा. ओके. फ्रॉम डाल दिया, फॉर्म डाल दिया. नहीं वो बाकी है. डॉक्यूमेंट्स सबमिट करके फी भी पे करना है ना. वैसे आदर्श तुम तो काफ़ी रिच हो ना, तुम कह रहे थे तुम मेरा फ़ी भर दोगे, तो हाँ 1700 भेज देना. ओके. यू आर डिप्रेस्ड ब्रो. इंडिड ब्रो. इसमें पूछने वाली कोई बात ही नहीं है. चलो पढ़ाई करते हैं अभी. जीपे नंबर एसजी. नंबर क्यों दूंगी भाई, क्यूआर कोड होगा ना उसके लिए. तो चलो पढ़ाई करते हैं. और मैं इसको साथ मजाक कर रही थी. ये लड़का कौन है, आयुष. गुड लक एवरीवन. अभी कोई भी कुछ भी नहीं लिखेगा. ओके. चैट सेक्शन पे मैं हाइड कर दे रही हूँ खुद के लिए. वी कांट सी इट. सेल साइकिल एंड सेल डिवीजन. सेल the continuous cell cycle. The continuous process of maintaining the cell with reference to chromosomal separation is referred to as cycle. The continuous process of maintaining the cell. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. Cell cycle and cell division. Cell cycle. The continuous process of maintaining the cell with reference to chromosomal separation is referred as cell cycle. So maintaining the cell with reference to chromosomal separation is referred to as the continuous process of maintaining the cell with reference to maintaining the cell with reference to chromosomal separation is referred as cell cycle. Includes preparatory phase referred as interphase. The phase represents the preparation for actual division to take place. Includes preparatory phase referred as interphase. The phase represents the preparation for actual division to take place. Then M phase include actual division of the cell. Preparatory phase referred as interphase. The phase represents the preparation for actual division to take place. The phase represents the preparation for actual division to take place. This is preparatory phase referred as interphase. M phase include actual division of cell. M phase include actual division of cell. Interphase accounts for more than around 95% of complete cycle. Interphase accounts for more than around 95% of complete cycle. Interphase accounts for more than around 95% of complete cycle, whereas M phase accounts for 5% of complete duration of cell cycle. Whereas M phase accounts for 5% of complete duration of cell cycle. 95% interphase, 5% M phase. Interphase accounts for more than around 95% of complete cycle, whereas M phase accounts for 5% of complete duration of cell cycle. Different cells have different duration. Human cell 24 hours yeast 90 minutes for cell division. Okay, human cells 24 hour, yeast 90 minutes. Division of the cell. The continuity of cell division. Preparation represents cell cycle. The continuity of cell division. The continuity of cell division. The continuity of cell division. Preparation represents cell cycle. The continuity of cell division. Preparation represent cell cycle. Continuity of cell division. Preparation represent the continuity of cell division. Preparation represents cell cycle. Cell cycle interphase. Preparatory phase, resting phase. Cell cycle interphase. Preparatory phase, resting phase. Actual cell division does not observe. Okay, preparatory phase or resting phase. Actual cell division not observed. Comprises of three different stages: G1 phase, S phase, and G2 phase. Subphases. Comprises of interphase consist of three phases: G1, S, and G2. G1, S, and G2. G0 phase. G1. G1 phase, gap 1 phase, most of the cell organelles duplication. Gap 1 phase, most of the cell organelles duplication. 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 RNA and protein synthesis. RNA and protein synthesis. RNA and protein synthesis. DNA precursor. Nucleotide, amino acid, sugar synthesized. Sugar synthesized. Longer the phase, slower will be the division. What is somatic cell? And diploid cell, somatic cells are diploid cell. G1 gap 1 phase. Most of the cell organelles duplicate in G1 phase. G1 phase, most of the cell organelles duplication. Most of the cell organelles duplicate. RNA and protein synthesis. Synthesis of RNA and protein. Most of the cell organelles duplicate. Most RNA and protein synthesis. DNA precursor. DNA precursor. Nucleotide, amino acid, sugar synthesized. DNA precursors. Nucleotide, amino acid, sugar synthesized. Longer the phase, slower will be the division. Longer the phase, slower will be the division. Most of the cell organelles duplicate herein. Most of the cell organelles duplicate here in the G1 phase. RNA and protein synthesis. DNA precursor. Nucleotide, amino acid, sugar synthesis. Longer the phase, slower will be the division. Longer the phase, slower will be the division. Duplication of organelles and synthesis of protein and RNA. S phase, synthetic phase, gap 1 phase, synthetic phase, DNA duplication, centriole duplication, histone protein, centrosome, centriole duplication, histone protein synthesis. S phase, synthetic phase, DNA duplication, centriole duplication, DNA duplication, centriole duplication, histone protein synthesis of histone protein, synthetic phase, DNA replication, DNA duplication, centriole duplication, DNA duplication, centriole duplication, and formation of or synthesis of histone protein. Gap 1 phase, chromosome X1 sister chromatid. S phase, DNA duplication, sister chromatid duplication. Okay, gap 1 chromosome sister chromatid, chromosome X 1 sister chromatid. In S phase, synthetic phase, DNA duplication, sister chromatid duplication, DNA duplication or sister chromatid duplication, DNA duplication, sister chromatid duplication. G1 phase. chromosome 1 sister chromatid duplication. 8 pg of S is equals to 16 pg. Is equals to 16 pg. If the total number of chromosomes at Z1 is X, total number of chromosomes at Z1 is X, number of chromosomes at S remains X. Total number of chromosomes at Z1 is X. Number of chromosomes at S remains. Total number of chromatid at Z1 is N. The number of chromatid at S is R2N. Total number of Z1 is 2C. Amount of DNA at S is equals to 4C. 2C 7 4C unsa 8 PC 7 G1 ma S ma 16 PC unsa double unsa. Okay. If the total number of chromosome at G1 is X, number of chromosome at S remains X. Number of chromosome will be same, but amount of DNA will change. The total number of chromosome X, here also X. Okay, one sister chromatid. DNA duplication, sister chromatid duplication. The amount of DNA increases. The total number of chromosome at Z1 X, number of chromosome at S remains X. Total number of chromosome at Z1 is N, then the number of chromatid at S is total number. Okay, number of chromatid increases, but number of chromosome remains same. If there is one chromosome, there will be one chromosome only, even after S phase. But number of chromatid and number of DNA, amount of DNA increases. The total number of chromatid at Z1 is N, then the number of chromatid at S is 2N. Okay, 2 to 4 N2. Okay. Number of chromatid and amount of DNA changes, but number of chromosome and amount of chromatid changes, but number of chromosome remains same. Phase most of the organelles duplicate, and then most of the organelles duplicate. Protein and RNA synthesis of protein and RNA occurs. Synthetic phase, DNA duplication, centriole duplication, histone synthesis of histone. G2 phase, gap2 phase, tubulin protein synthesis. Tubulin protein is synthesized. Tubulin protein is synthesized. Special set of organelles duplication. Chloroplast, mitochondria, Golgi bodies, RNA and protein synthesis. RNA and protein synthesize again in gap2. Okay. Special set of organelles duplication like chloroplast, mitochondria and Golgi bodies. Organelles, special set of organelles like chloroplast, mitochondria and Golgi body. Chloroplast, mitochondria and Golgi body duplicates here in G2 phase. Tubulin protein synthesis of tubulin protein, RNA and protein synthesize whatever it is. DNA precursor, nucleotide, amino acid, sugar synthesized. Longer the phase, slower will be the division. Tubulin protein synthesis, special set of organelle duplication, mitochondria, Golgi bodies and chloroplast. Chloroplast, mitochondria and Golgi body, RNA and protein synthesize. G0 phase, referred as quiescent phase. Okay. G0 phase, referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Z0 phase, referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. G0 phase, referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Metabolism observed. Metabolism observed. Z0 phase referred as quiescent phase. Phase of metabolism in the cell. Phase of metabolism in the cell without undergoing proliferation. Metabolism observed. Cell proliferation not observed. Metabolism observed. Cell proliferation is not observed. Okay. Phase of metabolism in the cell without undergoing proliferation. The condition where cell can exit the cell cycle. The condition where cell can exit the cell cycle temporarily in deficiency or requirement. Temporarily in deficiency or requirement, it appears into active cell division. It appears into active cell division permanently causes to divide nerve cells. Permanently causes to divide. Okay, temporarily in deficiency or requirement, it happens into active cell division. It happens into active cell division. Permanent ceases to divide. Okay, permanently ceases to divide. They are ceases to. They aim to divide. Nerve cell. Example, nerve cell. Example, nerve cell. Okay. G0 phase referred as quiescent phase. Phase of metabolism in the cell without undergoing proliferation. Metabolism observed. Cell proliferation not observed. The condition where cell can exit the cell cycle temporarily in deficiency or requirement. It appears into active cell division. Requirement in deficiency or requirement. It appears into active cell division. Permanently ceases to divide a nerve cell. Okay. G2 phase, formation of or synthesis of tubulin protein. Special set of organelles duplicate here like mitochondria, chloroplast and Golgi body. RNA and protein synthesis. G0 phase, quiescent phase. We will not take break, okay? कोई लिख दो वहां पर कमेंट पे ना, तो मैं पिन कर दूंगी. ये हम लोगों का ना 90 मिनट वाला स्लॉट 4 होगा. नाइन्टी फाइव का हो गया, कोई बात नहीं. 90 मिनट का स्लॉट स्टार्ट हो गया है. ओके, दिस इज आवर फ्लोट 4th स्लॉट. कोई लिख दो वहाँ चैट सेक्शन पर, मैं पिन कर दूंगी. G0 फेज रिफर्ड एज क्वेश्चन फेज. मैं लिख सकती हूँ लेकिन मुझे अभी ठंड लग रही है ना, मैंने जेब पे हाथ लगाया है. क्वेश्चन फेज, phase of metabolism in the cell without undergoing proliferation. Metabolism in the cell without undergoing proliferation. Metabolism observed. Cell proliferation is not observed. The condition where a cell can exit the cell cycle. Temporary permanent nerve cell. Amount of DNA increases, doubles, including chromatid. Chromatid also doubles, but chromosome it doesn't. Chromosome remains same. Number of chromatid is doubled. Mitotic phase, M phase, comprises of two process. Mitotic phase, M phase, comprises of two process: karyokinesis, cytokinesis. Mitotic phase, M phase comprises of two process: karyokinesis, nuclear division, karyokinesis, and cytokinesis. Cytokinesis, cell division. Thank you, Shubham. Comprises of two process: karyokinesis, cytokinesis, cell division, nuclear division, nucleus division. Karyokinesis, nuclear division. Okay, mitotic phase comprises of two process: karyokinesis and cytokinesis. Okay. Cell regulation. Cell cycle is regulated or maintained by certain signaling. Cell cycle is regulated or maintained by certain signalings. Signaling refers to the surveillance mechanism of the cell to check, stop, maintain the different phases in the cell cycle. Signaling refers to the surveillance mechanism of the cell to check, stop, or maintain the different phases in the cell cycle. To check, ensure the error-free process. Certain signaling, cell regulation. S phase, G1 phase, S phase, and G2 phase. We have quiescent phase, G0. M phase, mitotic phase, mitotic phase, mitotic phase. two process, karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Okay. Cell regulation is carried by checkpoints. Cell regulation is carried by checkpoints. Surveillance mechanism. Cell regulation is carried by checkpoints. Cell regulation. Okay. Majorly three checkpoints or surveillance mechanism. G1S checkpoint, G1 to S checkpoint, G2 to M checkpoint, G1 to S checkpoint, G2 to M checkpoint, and anaphase promoting complex, metaphase, anaphase transition, spindle assembly checkpoint, okay, G1 to S phase and G2 to M phase, G1 S checkpoint, G2 M checkpoint, anaphase promoting complex, anaphase promoting complex. Metaphase anaphase transition or spindle assembly checkpoint, okay, three checkpoints, G1 S checkpoint, G2 M checkpoint, and spindle assembly checkpoint. Spindle assembly checkpoint. Metaphase anaphase transition, anaphase promoting complex, G1S checkpoint, G2M checkpoint. Then we have prophase, metaphase, anaphase, telophase, and cytokinesis. Prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, first phase, prophase, metaphase, anaphase. Prophase, metaphase, anaphase, and prophase, metaphase, anaphase, telophase, and cytokinesis. Prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, it is between the anaphase and metaphase. Anaphase checkpoint, anaphase promoting complex. Okay, metaphase anaphase transition, metaphase anaphase transition, or anaphase promoting complex, anaphase promoting complex. These are the three checkpoints. Cell regulation is carried by checkpoints, surveillance mechanism. Cell cycle is regulated or maintained by certain signaling. Cell cycle is regulated or maintained by certain signaling. Signaling refers to the surveillance mechanism of the cell to check, stop, maintain the different phases in the cell cycle. To check, ensure the error-free process. Signaling refers to the surveillance mechanism of the cell to check, stop, maintain the different phases in the cell cycle. To check, ensure the error-free process. Error-free process बनाउनको लागि रहेछ. Okay. Surveillance, check, stop, maintain. Surveillance mechanism of the cell to check, stop, maintain the different phases in the cell cycle. Okay, first one is G1S checkpoint, G1S checkpoint. Then we have G2M checkpoint. Then we have between the anaphase and metaphase, between the metaphase and anaphase, spindle assembly checkpoint, Then we have anaphase checkpoint or spindle checkpoint, spindle assembly checkpoint. Metaphase anaphase transition, metaphase anaphase transition. G2M checkpoint. DNA synthesis and DNA replication are same term. DNA synthesis and DNA replication. Okay, DNA synthesis and DNA replication are same term. G1S checkpoint, first checkpoint, right? G1S checkpoint influenced by cell growth, DNA synthesis, precursor component, lack non-regulation. Okay, precursor component lack or non-regulation. Nutrients lack, referred as a restriction point, Once it is passed, cell cycle will complete. Restriction point. Once it is passed, cell cycle will complete. Cell growth, DNA synthesis. Precursor component. Lack, non-regulation. Nutrient lack, referred as restriction point. G2M checkpoint influenced by cell growth and DNA replication. It is influenced by cell growth and DNA replication. Anaphase, okay, DNA replication, DNA synthesis, but no replication, duplicate no. G1S checkpoint influenced by cell growth, DNA synthesis. Precursor component lack non-regulation. Lack of precursor component or non-regulation of precursor component. Nutrient lack, referred as restriction point. Restriction point. Once it is passed, cell cycle will complete. The Z2M checkpoint influenced by cell growth and DNA replication. APC, anaphase promoting complex, metaphase anaphase transition or spindle assembly checkpoint. APC influenced by microtubules formation in the lack of formation. Spindle assembly, lack of what? Spindle assembly. Attachment of spindle in kinetochore. No proper attachment. Checkpoints are regulated by cyclin dependent kinase, CDK, cyclin dependent kinase and cell division cycle kinase, CDC2. Transfer of phosphate bond energy, kinase, CDC2. Kinase is activated when it joins attached to cyclin. Two types of cyclins are observed. Kinase is activated when it joins attached to cyclin. Kinase is activated when it joins attached to cyclin. Two types of cyclin are observed. Z1 cyclin helps in G1S checkpoint associated with CDC2 kinase, CDK. Mitotic cyclin associated with CDC2K, C2 kinase forms maturation promoting factor. G2M checkpoint, mitosis. आया, what is this? इतना टफ था ना, इरिटेटिंग भी था, कुछ समझ में नहीं आया. Mitosis. Observed by Starberger in plant, Fleming in animals. Mitosis is observed by Starberger in plant and Fleming in animals. Term coined by Fleming. Okay, mitosis is the term coined by Fleming. Consists of two processes. Karyokinesis involves subphases. Cytokinesis involves two types of cell, animal cell and plant cell. Furrow formation in animal cell. Cell plate formation in plant cell. Karyokinesis subphases: prophase, metaphase, anaphase, telophase. Okay. Cytokinesis, karyokinesis and cytokinesis. Okay, karyokinesis subphase: prophase, metaphase, anaphase and telophase. Cytokinesis involve two types of cell, that is animal cell furrow formation, plant cell formation of cell plate. Mitosis, division of one cell into two daughter cell. Each daughter cell receives equal number of chromosome, which is equal number of chromosome in parent cell. Mitosis, division of one cell into two daughter cell. Each daughter cell receives equal number of chromosomes, which is equal number of chromosome in parent cell. Parent cell 46 chromosome, one cell. Okay, daughter cell 1, daughter 2, daughter 1, daughter 2, both receives 46 chromosome. Equal division, mitosis, equal division. Okay, division of one cell into two daughter cell. Each daughter cell receives equal number of chromosome, which is equal number of chromosome in parent cell. Generally absorbed in diploid animals and both haploid and diploid cell in plants. Generally absorbed, mitosis is generally absorbed in diploid cell of plant, sorry, animals, diploid cell of animals and diploid and haploid cell of plants. Both the diploid and haploid cell in plants generally absorbed in diploid cells in animals and both haploid and diploid cell in plants. Cardiokinesis, one daughter nuclei, two daughter nuclei. Cardiokinesis, one daughter nuclei is equals to two daughter nuclei in the same cell contains with in the same two daughter nuclei in the same cell contained within the cell. Prophase, first stage. Initial event of prophase, chromatin or chromosome condensation appears as entangled fiber. Centriole, centromere that was duplicated in the S phase of centromere that was duplicated in the S phase of interphase starts moving towards the pole. Centromere, centriole, I guess. Centrosome, centromere, centriole, centromere that was duplicated in S phase. In the S phase of interphase, it starts moving towards the pole. Elongated mature centriole at Z2. Astral rays formation. Emergence is observed in the initial prophase. The chromatin fiber appears like ball of wool, referred as spireme stage. Based on the astral formation, two types of mitosis. Without astral formation, anastral in plants, centriole absent, anastral, okay. Astral or amphiastral in animals, centriole present. With astral formation, I am kind of sleepy. थोड़ी देर सो जाती हूँ, काफी नींद आ रही है. Did I pause the timer? Yeah, I did. I'm sleepy. But I'll not sleep, okay? I'm kind of sleepy. Where is it? I'll wear socks. इतनी जल्दी? मुझे भी नींद आ रहा है लेकिन मैं सोने नहीं जाऊंगी। मैं इतना बड़ा बोतल में पानी भर के आऊंगी ना अभी, वो पीते पीते रहूंगी। लेकिन उससे पहले ना मैं इन लोगों की टेक्स्ट देख लेती हूँ। ये लोग ना कुछ कर रहे हैं, पता नहीं क्या करते रहते हैं ये लोग।
सजना, क्या तुम ये देख रहे हो? रोने का मन करे क्या? ब्रेकअप हो गया क्या सजना का?
ये मूवी देखा आज तो पहले प्यार याद आ गई। मतलब दूसरा अभी भी है। आई नहीं है इसलिए तो याद आया। याद करते रहो एग्जाम आ रही है। क्या करते रहते हैं ये लोग।
ब्रेकअप हो गया क्या सजना का? सजना, क्या तुम ये देख रहे हो? रोने का मन करे क्या? ब्रेकअप हो गया क्या सजना का?
दिस गर्ल्स रोशनी। हाय रोशनी, मैं अभी पानी भर के आती हूँ। हाँ, इतने बड़े बोतल में पानी भर के लाऊंगी ताकि मुझे नींद ना आए। Let me search Appy. अप्पी का नंबर नहीं है क्या मेरे पास? तो मैंने अप्पी का नंबर क्या लिख के सेव किया है? यूपीआई किया होगा शायद। हाँ तभी तो लग रहा था यूपी। नहीं, उसने चेंज किया ऐसा मुझे लगा। वो करिश्मा का था क्या? हाँ करी का तो था। हाय क्यों नहीं दिखाता है यहाँ पर? लीव इट, फिर मुझे नहीं देखना। मेरा हो गया, मेरा होगा, क्या होगा तुम्हारा? मैं तो पानी लेके आऊँगी। मेरा नहीं हुआ है अभी से, वो एक और चैप्टर बचा है, वो कंप्लीट करके ही सोऊंगी। अब पियेंगे ओके पानी, इतना सारा पानी पियेंगे कि नींद ही चला जाएगा। वैसे मुझे नींद आ रहा था लेकिन सो तो नहीं सकते। चलो मैं टाइमर ऑन कर देती हूँ फिर। डिलीट करने की ज़रूरत नहीं थी आदर्श, सब लोगों ने देख लिया है वहाँ पर क्या था। तुम्हारा कंप्लीट हो गया ना? चलो जिस जिसका नहीं हुआ है पढ़ लो, मेरा भी नहीं हुआ है। शायद 3 AM तक भी नहीं होगा, चांसेस तो कम ही है। वेयर इज माय रफ कॉपी? थैंक यू श्री। तुम पढ़ोगी नहीं क्या? पढ़ोगे नहीं क्या श्री? श्री तो लड़का है ना। डीपी चेंज कर लिया श्री ने, कोई बात नहीं। डीपी प्रोफाइल बोलते हैं शायद। How you doing, Ima? I'm fine, Roshni. How about you? Good luck all. यहाँ पर भी लिख देती हूँ गुड लक। अब पढ़ते हैं। Astral rays formation emerges. Same punch. Karyokinesis. Prophase. First is initial event of the prophase. Chromatin chromosome. Chromatid increase हो रहा था ना तब, और chromatin कहाँ, chromatid ही increase हो रहा था chromosome. Karyokinesis. One daughter nuclei, two daughter nuclei in the same cell, contained within the cell. Prophase. First stage. Initial event of the prophase. Chromosome condensation. Chromatin condensation appears as entangled fiber. Centriole. Centromeres that was duplicated in the S phase of interphase starts moving towards the pole. Parent centriole. Pericentriolar material. Daughter centriole. replication as phase. Okay, elongated matured centriole at G2. Elongated matured centriole at G2. Astral rays formation. Emergence is observed. Astral rays formation. Emergence is observed in prophase. Astral rays formation. In the initial prophase, the chromatin fiber appears like ball of wool, referred as spireme stage. Okay, in the initial prophase, the chromatin fiber appears like ball of wool, referred as spireme stage. Spireme stage. Initial prophase, chromatin fiber appears like ball of wool, spireme stage. It is known as spireme stage. Spireme stage. It is known as spireme stage. Spireme stage. Based on astral formation, two types of mitosis without astral formation, anastral in plants, centriole absent. Based on astral formation, two types of mitosis occurs with astral formation, astral, astral or amphiastral in animals, centriole present, anastral, absence of astral fiber, as in plants, centriole absent without the formation of astral rays. Spireme stays ball of wool. Spireme stays ball of wool. In the initial prophase, the chromatin fiber appears like ball of wool, referred as spireme stays. In the initial prophase, chromatin appear like ball of wool, which is known as spireme stays. Formation of astral rays, if present, it is as in animals, centriole present, astral or amphiastral. Without the formation of astral rays, anastral. In plants, centriole absent. Centriole is absent. Okay, centriole absent. Aster is composed of centrosome with astral rays. Aster is composed of centrosomes Aster is composed of centrosome with astral rays. Aster is composed of centrosome. Aster is composed of centrosome. Centriole, centromere. Aster is composed of centrosomes with astral rays. In total, two asters. The two asters associated with spindle fiber, microtubules. The two asters associated with spindle fiber, microtubules, collectively referred as mitotic apparatus. Collectively referred as mitotic apparatus. Collectively referred as mitotic apparatus. मैं भूल जाऊंगी ये वाला. Late events of prophase. Late events of prophase. Chromosomal condensation. Chromosomal condensation. Centriole movement polewards. Centriole movement. Centriole movement polewards. The astral rays formation radiation. The disappearance of nucleolus, ER, Golgi bodies, the disintegration of nucleus. The disintegration of nucleus. Okay. In the late prophase, centrosome or centriole, I guess. Centriole movement. Okay. Centriole move towards the pole. Chromosome is condensed. The astral rays formation occurs. The disappearance of nuclear membrane. The disappearance of nucleolus, endoplasmic reticulum, and Golgi bodies. Disappearance of nucleolus, endoplasmic reticulum, and Golgi bodies. Disintegration of nucleus. Prophase. At the completion of prophase, the chromosomal condensation. At the completion of prophase, the chromosomal condensation. Centriole, poleward movement, disappearance of nuclear envelope, disintegration. Disintegration of nucleus is simply disintegration of nucleus is simply a term for the disappearance of nuclear envelope. Disappearance of nuclear envelope is also called disintegration of nucleus. Nucleolus, ER and Golgi body disappears. Centriole moves towards the pole. Metaphase. After prophase there is metaphase, okay? It starts with all the chromosomes scattered or placed. It starts with all the chromosome scattered or placed, absorbed throughout the cell. Metaphase. It starts with all the chromosomes scattered or placed, absorbed through the cell. इसके बाद कौन सा photosynthesis, right? Photosynthesis in higher plants, plant growth and development. हाँ ये भी हो जाएगा, वो सब छोड़ देंगे, बाद में करेंगे. कितना रहेगा? 1 2 दो कर लिया तो कल 1 2 3 4 5 6 6 रहेगा ना. ये नीचे का 2 को नहीं छोड़ सकते, पहले भी नहीं किया है ना वो. फिर ये वाला स्टार्ट करेंगे. हाय देखते हैं. प्रोफेज. Metaphase. It starts with all the chromosomes scattered or placed, absorbed throughout the cell. Metaphase. It starts with all the chromosomes scattered or placed, absorbed throughout the cell. Initial metaphase. The chromosome short क्या लिखा है मैंने? The chromosomes and thickest. Shortest लिखा होगा, shortest लिख दिया. Short, SHORTEST, shortest. The chromosome shortest and thickest. Best phase to observe morphology of chromosome. Best phase to observe morphology of chromosome is metaphase. Best phase to observe morphology of chromosome. Centriole produce spindle fiber which connects the kinetochore. In metaphase, centriole produce spindle fiber which attach with kinetochore. Centriole produce spindle fiber which connects to kinetochore. Later metaphase, process of arrangement of chromosome. Metaphase plate. Metaphasic plate, equatorial plate arrangement. Equatorial plate arrangement, spindle fiber, spindle equator. The process of arrangement of chromosome in the metaphasic plate congregation. एक तो मेटाफेजिक प्लेट पे अरेंज होगा. मेटाफेजिक अरेंजमेंट, इक्वेटोरियल प्लेट अरेंजमेंट, स्पिंडल फाइबर और स्पिंडल इक्वेटर. स्पिंडल फाइबर connects to spindle fiber from centriole connects to kinetochore. The chromosome shortest and thickest. Chromosome are shortest and thickest. Best phase for the study of morphology of chromosome. The chromosome shortest, thickest, best phase to observe morphology of chromosome structure. Best phase to observe morphology of chromosome structure. Metaphase. Centriole produce spindle fiber which connects to kinetochores. Centriole produce spindle fiber which connects to kinetochore. The chromosomes shortest and thickest. ये initial metaphase, okay? It starts with all the chromosomes scattered or placed, absorbed throughout the cell. Initial metaphase. The chromosome shortest and thickest. Best phase to observe morphology of chromosome. Best phase to observe morphology of chromosome structure. Centriole produce spindle fiber which connects to kinetochore. Centriole produce, centriole ये बाहर वाला, produce spindle fibers which connects to kinetochores. Later metaphase, process of arrangement of chromosomes. Metaphase plate, okay? Later metaphase, process of arrangement of chromosomes, metaphase plate. Later, metaphase, process of arrangement of chromosome, metaphase plate. So arrangement of chromosome, metaphase plate, spindle fiber, spindle equator. The process of arrangement of chromosome in the metaphase plate is plate congregation. Okay, हो गया ये मेटाफेज में इक्वेटर, मेटाफेजिक प्लेट या फिर इक्वेटर में आके क्या होगा, स्पिंडल, स्पिंडल इक्वेटर में जाके अरेंज होगा. बेस्ट phase for the study, morphological study of chromosome. Spindle fiber attaches to, connects to kinetochore. हो गया. प्रोफेज, पहले तो क्या होगा, क्रोमोसोम बॉल ऑफ वूल जैसा लगेगा, इसको बोलते हैं अस्तगफिरुल्लाह, इसको बोलते हैं स्पाइरिम स्टेज. किससे भूल जाती हूँ, स्पाइरिम स्टेज, स्पाइरिम स्टेज, स्पाइरिम स्टेज. Astral, amphiastral, anastral, astral, amphiastral, animal cell, centriole present, anastral, centriole absent, plant cell. Two asters associated with spindle fiber, microtubules, collectively referred as mitotic apparatus. Mitotic apparatus, ये भूल जाएगी तू. Chromosome condensation, centriole movement, centriole moves towards the pole. लेट प्रोफेज में होता है ना ये, हाँ लेट प्रोफेज. सेंट्रियोल मूव टुवर्ड्स द पोल. एस्ट्रल रेज फॉर्मेशन रेडिएशन. ओके, डिसअपियरेंस ऑफ nucleolus, Golgi body and endoplasmic reticulum, disintegration of nucleus or nuclear envelope. Anaphase, prophase, metaphase and anaphase. Spindle fiber, aren't they? Yeah, they are. Centriole produce spindle fiber which connects to kinetochore. A spindle fiber are produced by centriole which connects to kinetochore. Best phase to observe morphology of chromosome structure. Best phase to observe morphology of chromosome. Who knows? April. Centriole produce spindle fiber which connects the kinetochore. अब क्यों सोच रही है? Don't think about it. Anaphase, upstage. Anaphase, upstage. The separation of sister chromatids towards each pole. The separation of the separation of sister chromatid towards each pole. Separation is brought by shortening of spindle fiber. Shortening of spindle fiber connected to kinetochore and elongation of interzonal fiber. The separation of sister chromatid towards each pole. Upstage, anaphase. Upstage, ana means up, ana up. Phase stage, the separation of sister chromatid towards each pole. The separation of sister chromatid towards each pole. Separation is brought by shortening of spindle fiber connected to kinetochore. Separation is brought by shortening of spindle fiber. Separation is brought by shortening of spindle fiber connected to kinetochore. The separation of sister chromatids हाया the separation of sister chromatid क्यों ऐसा हो रहा है? Don't think about it, don't think about it. The separation of sister chromatid towards each pole, दोनों pole में separation of sister chromatid. Separation is brought by shortening of spindle काइनेटोकोर और सेंट्रियोल के बीच में लगा था. सेंट्रियोल से प्रोड्यूस होकर काइनेटोकोर से अटैच था वो. शॉर्टनिंग ऑफ स्पिंडल फाइबर कनेक्टेड टू काइनेटोकोर एंड एलोंगेशन ऑफ इंटरजोनल फाइबर. एलोंगेशन ऑफ इंटरजोनल फाइबर. एलोंगेशन ऑफ इंटरजोनल फाइबर. ओके यू centromere division towards the pole side. Division towards the pole side. Centromere division towards the pole side. Centromere division. यार इसको पॉज ही कर दिया था. Centromere division towards the pole side. Chromatids are trailing behind. Centromere division towards the pole side. Chromatids are trailing behind. Centromere division towards the pole. Centromere division towards the pole. Chromatids are trailing behind. Shortening of spindle fiber. Interzonal fiber. Shortening of spindle fiber. Interzonal fiber क्या हो रहा है? Elongation of interzonal fiber. Elongation of interzonal fiber. ये एनाफेज की बात है. ओके. Shortening of fiber, elongation of interzonal fiber. The separation of sister chromatid towards each pole. Separation is brought by shortening of spindle fiber connected to kinetochore and elongation of interzonal fiber. Interzonal fiber. Elongated. Shortening of spindle fiber. Centromere division is towards the pole. Centromere division is towards the pole. ये मेरे मुंह में आ रहा है ऐसे ठुस ठुस करके. मुझे इतना गुस्सा आ रहा है ना इसके ऊपर, लेकिन मैं कुछ नहीं कर सकती. ठंड भी तो है. Centromere division towards the pole side. मैं इसको थोड़ा सा यो कर देती हूँ ना, खोल देती हूँ ना, क्योंकि ये ना ऊपर आके मुझे ऐसे ठुस ठुस कर रहा है. ठुस नहीं, ये मेरे ना चिन पे पता नहीं अनकंफर्टेबल फील करवा रहा है. Centromere division towards men. ठुस बोल दिया. Centromere division towards the pole side. Centromere division towards the pole side. Hi Salman, but now don't text. We can't talk right now. This is the study time and we have to study. So please don't text. Centromere division towards the pole side. Chromatids are trailing behind. Interzonal fiber. Elongation of interzonal fiber. Shortening of spindle fiber. Division of centromere is towards the pole. Chromatids are trailing behind. These are the condition of anaphase. This is separation of sister chromatids towards each pole. Okay. As each chromatids are separated from the chromosome, the chromatids aggregate at each pole, becomes the part of daughter nuclei. I was 21 that time and I didn't realize that ये भी होने वाला है. कोई बात नहीं. As each chromatids are separated from the chromosome, वो नहीं होना था ना, लेकिन हो गया, क्या ही कर सकते हैं. As each chromatids are separated from the chromosome, the chromatid aggregate at each pole becomes the part of daughter nuclei. Becomes part of daughter nuclei. As each chromatid are separated from the chromosome, as each chromatid are separated from the chromosome, the chromatid aggregates at each pole. As each chromatids are separated from the chromosome, the chromatid aggregate at each pole becomes part of daughter nuclei. Becomes part of daughter nuclei. Considering each chromatid is equals to one chromosome. Each chromatid is considering each chromatid equals to one chromosome. Explanation: human cell is equals to 46 chromosome, right? Z1 46, S 46 chromosome. chromosome तो चेंज नहीं होला है. Chromatid equals to 46, तो यहाँ तो डबल होगा ना. Chromatid 46 into 2 92. G2 G1 G2 G2 46 chromosome, 92 chromatids. Prophase same, metaphase same, anaphase 92. Okay, 46 chromosome for one daughter, 46 chromosome for other daughter. But chromatid 46 chromosome and 92 chromatids. 92 chromatid is divided into 46 chromosome and 92 chromatid is divided into two set, that is 46 chromatid and 46 chromatid. Anaphase, exactly same number of chromatid as Z2. Exactly same number of chromatid as G2, double number of chromosome as G2. G2 में 46 क्रोमोसोम है, एनाफेज में तो 42 ही है. डबल नंबर ऑफ क्रोमोसोम एज G2. क्या लिखा है मैंने ये? As each chromatids are separated from the chromosome, as each chromatids are separated from the chromosome, as each chromatids are separated from chromosome, the chromatid aggregates at each pole becomes the part of daughter nuclei. Considering each chromatid equals to one chromosome. Each chromatid, one chromatid equals to one chromosome. Okay, 46 chromosome. G1 phase 46 chromosome. S phase there 46 chromosome. But chromatid 46 to 92. 92 chromatid, 46 chromosome, right? S phase. Number of chromatid increases from G2. 46, 92 chromatid. Prophase same, metaphase same, anaphase 46 chromosome, and chromatid is again distributed to two daughters, that is 46 set for each. 46 chromatid, okay. Anaphase, exactly same number of chromatid as G2. Double number of chromosome as G2. Chromosome double, okay. 92 chromatids. Telophase, reverse of prophase. Telophase is reverse of prophase. Chromosome, chromatin, decondensation of chromosome, centriole disappear, spindle fiber disappear, nuclear membrane reappears, nucleolus, and Golgi body reappears, nuclear membrane reappears, nucleolus, and Golgi body reappears, centriole disappear, spindle fiber disappears. अच्छा, spindle fiber disappears. Spindle fiber disappears. reverse of prophase. Okay, chromosome, chromatin, decondensation. Centriole disappears, spindle fiber disappears, nuclear membrane reappears. Nucleolus, ER and Golgi body reappears. They all reappears. Nucleolus, ER, Golgi body reappears. Nuclear membrane reappears. Centriole disappears, spindle fiber will disappear. Centriole and spindle fiber disappears. Chromosomes, that is chromosomes, chromatin, decondensation. Okay, chromosomes, chromatin, decondensation. Decondensation of chromosome or chromatin. Reverse of prophase, chromatin, decondensation. Chromosome, chromatin, decondensation. अनवन आज कहीं से आ गया है. Late telophase, cytokinesis, cell division, plant cell, animal cell, foro formation in animal cell, cell इसमें plate formation होगा ना, cell plate, plate formation in plant. Animal cell, foro formation, construction, microfilament accumulated at the central body of the cell. Microfilament accumulated at the central body of the cell referred as mid-body. The actin and myosin filament starts pulling. The actin and myosin filament starts pulling the wall side. The actin and myosin filament starts pulling the wall side. Formation of constriction. Formation of constriction towards the center. Centripetal fashion. Okay, mid-body. Accumulation of microfilament, mid-body. Accumulation of microfilament, mid-body. Formation of constriction. Foro formation, construction, microfilament accumulated at the central body. We don't have to think about it. Microfilament accumulated at the central body of the cell referred as mid-body. Accumulation of microfilament, mid-body. Foro formation, construction. Okay, microfilament accumulation at the central body of the cell referred as mid-body. Accumulation of microfilament, mid-body. The actin and myosin filament starts pulling the wall side. The actin and myosin filament starts pulling. Actin filament, microtubules. Microfilament. Okay, microfilament accumulated at the central body of the cell referred as mid-body. The actin and myosin filament starts pulling the wall aside. Of constriction towards the center. Formation of constriction towards the central. Centripetal fashion. Okay, centripetal fashion. Alag cell. Cell plate formation, plant cell. The vesicles are produced from Golgi bodies. The vesicles fuse together to form phragmoplast. The vesicles fuse together to form phragmoplast. Also, the ER vesicle, endoplasmic reticulum vesicle, contribute to phragmoplast. Formation of vesicles. Vesicles are produced from Golgi bodies. Formation of vesicles from Golgi bodies. The vesicles fuse together to form phragmoplast. Vesicles from Golgi bodies. Formation of vesicles from Golgi bodies. Vesicles are produced from Golgi bodies. The vesicles fuse together to form phragmoplast. The vesicles fuse together to form phragmoplast. Also, the ER vesicle contribute to endoplasmic reticulum vesicles contribute to phragmoplast. Cell plate, plant cell, cell wall. Cell plate, plant cell, cell wall. Vesicle. Foro or constriction is not possible in plant cell. Okay, vesicle, microtubules along with microtubules fuse together to form phragmoplast. Okay, phragmoplast, then cell plate formation, plant cell, cell wall. The microtubules are also fused with phragmoplast, starts forming cell plate. Precursor of cell plate is phragmoplast. Towards the periphery, starts forming cell plate towards the periphery. Centrifugate. Okay, किती centripetal थी animal ना, centripetal fashion. Okay, centripetal fashion in animal, centripetal. Now centrifugate pattern result into calcium ion, magnesium pectate deposition. Calcium and magnesium pectate deposition results into middle lamella. cell separation, middle lamella cell separation, leads result into calcium and magnesium pectate deposition, calcium and magnesium pectate deposition, centrifugate pattern towards the periphery, centrifugate pattern, precursor of cell plate is phragmoplast, precursor of cell plate is phragmoplast. The microtubules are fused with phragmoplast, starts forming cell plate towards the periphery, centrifugate pattern results into calcium and magnesium pectate deposition, result into middle lamella cell separation. Important point: mitosis. Mitosins, the chemical substances which induces the mitotic division, cell division. Okay, mitotic division, the chemical substances which induces the mitotic division. These are known as mitosins. Mitosins, auxins, cytokinin, gibberellin, and insulin. Mitosins, the chemical substances which induces mitotic division, the chemical substances which induces mitotic cell division, mitotic induces mitotic division, cell division. Okay, these are gibberellin, cytokinin, auxin, and insulin. Insulin, gibberellin, cytokinin, and auxin. ACG, ACGI, GI, ACGI, ACGI, auxin, cytokinin, gibberellin, and insulin. ऑक्सिन साइटोकाइनिन जिब्रेलिन एंड इंसुलिन, ये तो मैं भूल ही जाऊंगी. माइटोजेंस इसको लिखो ना. नहीं अभी नहीं, दूसरे में एक बार ही कर लेती हूँ, फिर लिखती हूँ. अय्या ऑक्सिन, सिग्निफिकेंस ऑफ माइटोसिस, फिर मियोसिस, इसके बाद तो कोई करना खत्म. रिजल्ट्स इनटू मिडिल लैमेला. हाँ वो वाला एक बार और अच्छे से करते हैं ना, कल फिर यही 3 कर लेते हैं. अगर तुमने आज प्लांट ग्रोथ एंड डेवलपमेंट किया तो अच्छी बात है. प्लांट ग्रोथ एंड डेवलपमेंट हो जाएगा एक और. बाकी सब छोड़ दो वहाँ से. बायोमोलिक्यूल्स से, हाँ ये 3 अच्छे से करो ना, और ये वाला फिर न्यूरल वो करना है. लोकोमोशन एंड मूवमेंट भी हो गया, एक्सक्रीशन वाला भी हो गया, फ्लूइड बॉडी फ्लूइड एंड सर्कुलेशन भी हो जाएगा, ये भी हो जाएगा. ये न्यूरल कंट्रोल एंड कोऑर्डिनेशन, केमिकल कोऑर्डिनेशन एंड इंटीग्रेशन, ये 3 एक ही साथ करोगे तो अच्छा रहेगा. नहीं मैं बहुत सारा छूट जाएगा शायद. टुडे इज 17, 18, 19, 20, 10 दिन ही तो है. 10 से ज़्यादा चैप्टर्स बचे हैं. Centrifugal pattern, centrifugate pattern, okay, leads result into calcium 2 plus, Mg2 plus pectate deposition, results into middle lamella, cell separation. Mitosis, important point, mitosis, important point, mitosis. Mitosins, the chemical substances which induces the mitotic division, cell division. Auxin, cytokinin, gibberellin, and insulin. Auxin, cytokinin, gibberellin, and insulin. Auxin, cytokinin, gibberellin, and insulin. Hiya! Mitotic poison, the substance which suppresses half the division, the substance which suppresses half the division. Example: azide, cyanide, mustard gas, agglutination, chromosome grouping, colchicine. Check the separation by stopping the polymerization of spindle microtubule. Allow polyploidy. 2x, 4x, 46, 92, 92 क्रोमोसोम. ओके, 8, 16 क्रोमोसोम ही चेंज हो रहा है. माइटोटिक पॉइजन नहीं दिया है ना यहाँ पर ना. इट्स नॉट गिवन. सिग्निफिकेंस ऑफ माइटोसिस दिया है. माइटोटिक पॉइजन, the substances which suppresses half the division, azide, cyanide, mustard gas, and colchicine. Checks the separation by stopping the polymerization by checking or stopping the polymerization of spindle microtubules. शायद जाएंगे लेकिन अभी नहीं, फिर कभी ईयर के एंड में शायद जा सकते हैं, है ना? इनफ ही होगा। 3 इंटू 10 किया जाए तो 30 तो यहीं से हो जाएगा ना, आई थिंक। 30 हाँ, लगभग 30,000 होगा। अजाइड साइनाइड मस्टर्ड गैस agglutination chromosome grouping. Okay, mitotic poison, the substance which suppresses half the division, azide cyanide mustard gas, agglutination chromosomes grouping, chromosome grouping. Colchicine checks the separation by checking stopping the polymerization of spindle microtubules, allow polyploidy. By checking stopping the polymerization of spindle microtubules, allow polyploidy. The colchicine, natural mitotic poison obtained from colchicum. colchicum ही है ना ये, मैं प्रोनाउंस तो गलत नहीं कर रही हूँ ना? लिलिएस, पता नहीं मैं क्या पढ़ रही हूँ, लेकिन मुझे लग रहा है कि मैं सही पढ़ रही हूँ। mitotic poison, the substance which suppress half the division, which suppress or halves the division. Azide cyanide mustard gas, agglutination chromosome grouping. Colchicine polyploidy, 2x 4x polyploidy. Okay, if there are 46 chromosome it will become 92. If there are 8 chromosome it becomes 16. The number of chromosome is also getting doubled. Check the separation by checking stopping the polymerization of spindle microtubule. Allow polyploidy. The colchicine, natural mitotic poison obtained from colchicum. The colchicine, natural mitotic poison obtained from colchicum, liliase. Significance of mitosis. ये मुझे समझ में नहीं आया, मैं इसको फिर से करूँगी, वहाँ से मॉड्यूल से करूँगी इसको। Significance of mitosis. Growth and development of organism, zygote multicellular organization. The growth and development of organisms, zygote multicellular organization, maintains nucleocytoplasmic ratio. Increase in case of growth of cell, helps in maintaining the proportionate component of both. Maintain the surface area to volume ratio. पता नहीं क्या लिखी हूँ मैं, गुस्सा ही आ जाता है मैंने, लेकिन कर ही क्या सकते हैं पढ़ने के अलावा। मियोसिस, हाय, जो इजी पार्ट है ना मुझे उसमें ही प्रॉब्लम आ रहा है, तो मैं जो मुश्किल पार्ट है वो कैसे करूँगी, खुद ही सोचो तो। माइटोटिक पॉइजन, द सब्सटेंस विच सप्रेस हाफ द डिवीजन। ओके, ये हो गया। सिग्निफिकेंस ऑफ माइटोसिस, द ग्रोथ एंड डेवलपमेंट ऑफ ऑर्गेनिज्म, growth and development of organism, zygote multicellular organization, maintains nucleocytoplasmic ratio. nucleocytoplasmic ratio मतलब in case of growth of cell, helps in maintaining the proportionate component of both. Maintains the surface area to volume ratio. surface area volume high, surface area volume low. डिवाइड हो गया। surface area volume increases, perform surface area volume जत्ती वो ज़्यादा perform efficiently, perform efficiently। अच्छा ये है, ठीक है। maintains chromosomal number, maintaining the identity of, helps in repair and maintenance, example lining of guts. Maintains chromosomal number, maintaining the identity of, helps in repair and maintenance, example lining of guts. Numerical problems related to mitotic division concept. 1 से 2 हो गए ना T1 में, तो 2 से 4 होगा T2 में। T1 plus T2 40 minute, 1 division is equals to 20 minute. 40/20 t upon 1 division time. आई क्या किया है मैंने ये T1 प्लस T2 दो ओके एक डिवीजन में 20 मिनट लगता है तो दो डिवीजन में 20 मिनट ही लगे 20 प्लस 20 40 मिनट लगेगा ना। एक डिवीजन में 20 मिनट तो 40/20 इज इक्वल्स टू T अपॉन 1 डिवीजन टाइम वो कितना जनरेशन होगा ये निकलेगा नंबर ऑफ जनरेशन 2 calculation of total daughter cells after time t if the time of one division is given. If time of one division is 15 minute, total number of daughter produced after 150 minute. 150 मिनट की बात। अच्छा एक डिवीजन 105 मिनट, एक डिवीजन के लिए 15 मिनट, तो 5 1 जा 5 इंटू 2 ही तो करना है ना। 5 1 जा 5 5 3 जा 15 5 2 जा 10 5 1 जा 5 3 1 जा 3 3 7 जा 21 7 जनरेशन गया है ना। तो 7 इंटू 2 करना है 7 1 7 7 14 आई 14 तो नहीं होगा, इससे तो ज़्यादा ही होगा। जनरेशन तो 7 है, वो 2 टू द पावर करना है। गॉट इट 2 टू द पावर 7 7 7 7 14 आइया फिर से 7 आ गए। 2 2 4 1 4 4 2 8 8 1 8 8 16 16 1 16 16 2 अच्छा अब नहीं आएगा ना। 2 2 4 4 2 8 8 2 16 2 2 4 4 2 8 8 2 16 16 2 32 1 2 3 4 5 6 7 2 2 4 4 2 8 8 2 16 16 2 32 32 2 60 64 64 2 64 2 8 8 8 यहाँ से 2 आया 7 6 12 13 14 148 इज इट 148 60 60 148 ही ना 60 60 लग तो नहीं रहा 28 128 होगा ये तो 2 4 जा 8 राइट 8 2 6 से 12 हाँ 128 होगा 148 नहीं। तो टोटल 128 डॉटर निकलेगा सेल्स। फिर अब आएगा कैलकुलेशन ऑफ डॉटर टोटल डॉटर सेल आफ्टर टाइम टी इफ द टाइम फॉर वन डिवीजन इज गिवन। ओके इफ द टाइम फॉर वन डिवीजन इज 15 मिनट टोटल नंबर ऑफ डॉटर प्रोड्यूस आफ्टर 105 मिनट इज इक्वल्स टू 105 डिवाइडेड बाई 15 जिसमें से 7 निकलेगा तो टोटल 7 जनरेशन गया है 7 generation n equals to 7 that is 7 generation. Total number of daughter cell is equals to 2 to the power n 2 to the power n that is 2 to the power 7. Number of generation how many times same thing okay. तो यहाँ पर तुम निकाल सकती हो ना एक के लिए ये लोग 20 मिनट लगा रहे हैं तो दो के लिए कितना होगा 40 होगा। 21 से 20 22 से 40 तो यहाँ पर इन लोगों का सेकेंड जनरेशन चल रहा है 2 to the power n 2 to the power 2 तो डॉटर जो प्रोड्यूस होगा वो 4 प्रोड्यूस होगा। अच्छा इस जनरेशन में 4 होगा ना 1 2 3 4 हाँ तो वो भी उस जनरेशन की बात कर रहे हैं जहाँ पर ये होगा 126 सेवेंथ जनरेशन में 126 होगा आई 128 4 होगा इसमें 8 होगा इसमें 16 होगा इसमें 32 होगा इसमें 64 होगा हाँ इसमें 128 होगा। I think now you can do it. What is the total number of generation of cell division is what is the total number of what is the total number of generation of cell division is observed during mitotic division when the total daughter cell produces 64 total okay 64 है ना। What is the total number of generation कितना जनरेशन होगा अगर 64 है तो डॉटर सेल 2 to the power कितना is equals to 64 I think 2 2 is 4 4 is 4 4 2 is 8 8 1 is 8 8 2 is 16 16 1 is 16 2 is 32 32 into 32 हाँ 1 2 3 2 2s are 4, 1s are 4, 4 2s are 8, 8 1s are 8, 8 is 16, 16 1s are 16, 16 2s are 32, 32 2s are 64. तो 1 2 3 4 5 6, ये 6th जनरेशन होगा तुम्हारा. 2n is equals to 64 है ना. 2 2s are 4, 1s are 4, 4 2s are 8, 8 1s are 8, 8 is 16, 16 1s are 16, 16 32. अरे यहाँ पर मैंने क्यों लिखा 26, ये 64 होगा भाई साहब. 26 लिख दिया, 64 होगा. 64 को 2 to the power 6 लिखेंगे. 1 2 3 4 5 6. हाँ अब सही है ना. ओ अच्छा 2 to the power 6 लिखा था. सही है सही है. यहाँ पर 64 तो 2 to the power 6. एंड जनरेशन. इफ आस्क्स अबाउट टोटल नंबर ऑफ माइटोटिक डिवीजन, कितना माइटोटिक डिवीजन हुआ? तो 4 - 1 टोटल नंबर ऑफ माइटोटिक डिवीजन. 1 2 3 4 5 6 7. 4 - 1 is equals to 3 division. टोटल नंबर ऑफ डॉटर सेल इक्वल्स टू x. ये फिर क्या खिचड़ी पकाया है. एक बार mitotic division, how many times? Leaves us 61. Total number of generation. टोटल नंबर ऑफ माइटोटिक डिवीजन. ये जो इम्पोर्टेंट है वही वाला ही मैं भूल गई. वाह क्या बात है. टोटल नंबर ऑफ डॉटर सेल. डॉटर सेल जितना है उसमें से एक माइनस करो ना. तो यहाँ पर ओके. टोटल नंबर ऑफ डॉटर सेल x. नंबर ऑफ माइटोटिक डिवीजन x - 1. डॉटर सेल -1. अच्छा 16 डॉटर सेल है तो -1. 15 होगा तो 15 माइटोटिक डिवीजन. ऐसा भी हो सकता है क्या? टोटल नंबर ऑफ माइटोटिक डिवीजन 4 - 1 3 डिवीजन. 4 सेल रिड्यूस हुआ है. टोटल नंबर ऑफ डॉटर सेल इक्वल्स टू x. नंबर ऑफ माइटोटिक डिवीजन x - 1. टोटल नंबर ऑफ डॉटर सेल -1. नंबर ऑफ माइटोटिक डिवीजन. नंबर ऑफ माइटोटिक डिवीजन इक्वल्स टू माइटोटिक डिवीजन. नंबर ऑफ total number of mitotic division. Number of mitotic division equals to x - 1, where x is daughter cell. Total number of daughter cell, where x is total number of daughter cell. Total number of daughter cell. If asks about total number of mitotic division, -1 3 division. 3 division. आज ही तो था संडे. क्या 5 होगा? आंसर में तो सर ने 3 लिखाया है. 4 - 1. 4 - 1 तो 3 ही होगा ना ऑब्वियसली. cork cell robot hook. Cork cell. Dead cell. Okay. Cell cycle and cell division. Division. Number of daughter cell -1. Cell minus. Number of daughter cell -1. Number of daughter cell -1. Are you aware that all organisms, even the largest largest starts their life from single cell. You may wonder how a single cell then goes on to form such large organism. Growth and reproduction are characteristics of cell. Indeed, of all living organism, all cell reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of single parental cell. And its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of cell, DNA replication and cell growth also take place. During the division of cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome synthesize the other constituent of the cell and eventually divides into two daughter cells is termed cell cycle. Okay, the sequence of events by which cell duplicates its genome. The sequence of event by which cell duplicates its genome synthesize the other constituent of the cell. I don't think about it, Ima. Shh. The sequence of event by which a cell duplicates its genome synthesize the other constituent. इतना गुस्सा आता है ना, लेकिन कोई बात नहीं, छोड़ दो. Synthesize the other constituent of cell and eventually divides into two daughter cell is termed as cell cycle. Cell duplicate its genome. Okay, synthesis of other constituent of the cell and eventually divides into two daughter cells is termed as cell cycle. के cell cycle के लिए एक तो genome को duplicate करना पड़ेगा, फिर जो important cell organelles है उसको synthesize करना पड़ेगा, eventually divides, उसके बाद ही division होगा. इसे ही क्या बोलते हैं, cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, cell growth, DNA synthesis occur only during one specific stage in the cell cycle. The replicated chromosomes DNA, okay. The replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. The replicated chromosomes DNA are then distributed to daughter nuclei by complex series of events during cell division. These events are themselves under genetic control. The replicated chromosome DNA are then distributed to daughter nuclei. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. यू नो, तुम सबको पता है, एक मिनट टाइमर पॉज करके बोल लेती हूँ ना. जो एक ना मच्योर आदमी होता है ना, जो मेरी एज की होती है और जो इनविंसिबल जो भी है, उसकी एज के इंसान होता है ना, उन दोनों में क्या फ़र्क होता है? किसी को पता है? अगर नहीं पता है ना तो मैं बता देती हूँ जल्दी जल्दी में, क्योंकि मुझे पढ़ना भी है ना। जो मेरे एज का इंसान है ना, उसको दिखाना नहीं है, दिखावा नहीं करना है ना। लेकिन जो इनविंसिबल के एज का है ना, उसे पता नहीं क्यों दिखाना है। ये छोटे लोग होते हैं ना, लाइक यंगर जनरेशन, इन लोगों को हमेशा कुछ ना कुछ दिखाना होता है कि मुझे सब कुछ आता है। आई नो भाई तुझे आता है, लेकिन फिर वो फिर क्वेश्चन पूछ के ना ऐसे करके तुम्हें टेस्ट करेंगे कि तुम्हें आता है कि नहीं, सिर्फ उसे ही आता है, ऐसे करेंगे। लाइक ग्रो अप किड। मैं ऑलरेडी उस चीज़ के लिए बुढ़ी हो चुकी हूँ। आई थिंक मैं तुम्हारे एज से ही पास ऑन होके ना अभी इतनी बड़ी हो गई हूँ, लेकिन आई थिंक मैंने ना ऐसा छपड़ी बनती तो नहीं किया होगा उस टाइम, या फिर किया होगा। आई डोंट थिंक मैंने ऐसा किया होगा। मैं कभी दिखावा नहीं करती थी तब। पानी पी ही लेती हूँ। मुझे एक और स्टोरी याद आ गया। मैं जब क्लास 10th में थी, वो हम लोगों के साथ ही पढ़ता था। ओके, लाइक ही इज़ नॉट नेपाली, वो ना बिहार का है शायद, बट आई एम नॉट श्योर उसका, क्योंकि ना सरनेम के पीछे शाह था। और जो भी ये शाह लोग होते हैं ना, वो बिहार से ही आते थे। तो पता नहीं वो ज़्यादा छपरी था कि क्या था, मुझे नहीं पता। लेकिन वो क्लास 10 तक ही मेरे साथ पढ़ा है। फिर मैं दूसरे स्कूल गई और वो एक और स्कूल गया था। तो वहाँ पर मेरी फ्रेंड भी थी ना, तो उसने मुझे बोला कि उसने बाहर निकल के ना सारे उसको अपने दोस्तों को बोला कि उसने एग्ज़ाम में सब कुछ कर लिया है, और उसने बोला कि वो ही टॉप करेगा इस बार। ऐसा करके सारे दोस्तों को बोल दिया। फिर बाद में आई थिंक वो फेल तो नहीं हुआ, लेकिन बहुत कम नंबर आया उसका। मेरी पढ़ाई से आपको क्या लेना देना? वैसे भी मैं स्टोरी बोल रही हूँ अभी। मुझे आई थिंक आजकल ना आदत हो गई दूसरों की ना प्रोफाइल विजिट करने की। थैंक्स शुभम, टाइम आउट देने के लिए। आई एम जस्ट हेल्पिंग। थैंक यू सो मच। नहीं, लेकिन मैंने अपने एंड से बताया कि ऐसा सिचुएशन हुआ था। और जब हम क्लास 10 में भी थे ना, तो वो ऐसा बोलता था कि वो सबसे ज़्यादा स्कोर करेगा मैथमेटिक्स में, वो सबसे ज़्यादा स्कोर करेगा। आई डोंट नो, वो खुद के लिए बोलता था कि सबको दिखाने के लिए बोलता था, लेकिन उसने सबको बोला कि वो मैथमेटिक्स में सबसे ज़्यादा स्कोर करेगा। हालांकि उसने नहीं किया। तब कोई एक और था, अनिल गुरुंग करके, एक नेपाली ही था लड़का, उन्होंने किया था। ज़्यादा नहीं था, लेकिन हाँ उन्होंने टॉप किया था। अब हम लोग ना, वो दो मेरे कॉलेज में ही था। वो फिजिक्स डिपार्टमेंट में था, मैं केमिस्ट्री में थी, लेकिन फिर भी पता नहीं हम लोग बात नहीं करते थे। अभी भी मैं मिलती हूँ कभी कभी बैंक में, लेकिन मैं ऐसे ना देखी जैसे एक्टिंग करती हूँ। लीव इट, क्योंकि वो अब मैटर नहीं करता ना। The sequence of events by which a cell duplicates its genome, synthesizes the other constituent of the cell, and eventually divides into two daughter cell is termed as cell cycle. Eventually divides into two daughter cell is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in life cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phase of cell cycle. A typical eukaryotic cell cycle is illustrated by human cell in culture. A typical eukaryotic cell is illustrated. A typical eukaryotic cell cycle. Eukaryotic cell cycle. Cell cycle is A typical eukaryotic cell cycle is illustrated by human cell in the culture. These cells divide once in approximately about 24 hours. A typical eukaryotic cell cycle is illustrated by human cell in culture. These are human cells are divided once in approximately every 24 hours. However, the duration of cell cycle can vary from organism to organism, from cell type to cell type. The duration of cell cycle can vary from organisms to organism and also from cell type to cell type. Organism to organism and from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases. Okay, yeast for example can progress through the cell cycle in only about 90 minutes. हाँ वो काफ़ी भयानक रहेगा लेकिन क्या कोई नहीं कुछ नहीं कर सकते. Yeast for example can progress through the cell cycle in only about 90 minutes. Yeast, for example, can progress through cell cycle in only about 90 minutes. For example, can progress through the cell cycle. It's okay. Human 24 hour, okay. The cell cycle is divided into two basic phases, interphase and M-phase, mitosis phase. Interphase and mitosis phase. The M phase represents the phase when the actual cell division or mitosis occur. M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phase. Interphase represents the phase between two successive and M phase. Represents the phase between two successive M phase, and interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle of human cell division proper lasts for only about an hour. The M phase represents the phase when the actual cell division or mitosis occur. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phases. The M phase represents the phase when the actual cell division or mitosis occurs. It is significant. It is significant to note that in the 24-hour average duration of cell cycle of human cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. Duration of cell cycle. The M phase starts with the nuclear division. The M phase starts with nuclear division. The M phase starts with the nuclear division corresponding to the separation of The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. The M phase starts with the nuclear division. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in orderly manner. I don't think about it, bro. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, karyokinesis. And usually ends with the division of cytoplasm, cytokinesis. Separation of daughter chromosome. The interphase, though called the resting phase, interphase is also called resting phase, is the time during which the cell is preparing for division or by undergoing both cell growth and DNA replication. Interphase में क्या cell growth and DNA replication. In orderly manner, cell growth, DNA replication. The interphase is divided into three further phases. Gap 1, G1 phase, S phase, that is synthetic phase, and G2, gap 2 phase. Gap 1, synthesis, and gap 2 phase, okay? Cell grows in size. Cell grows in size in G1 equal to okay. G1 phase corresponds to the interval between mitosis and the initiation of DNA replication. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Between mitosis and initiation of mitosis and M phase, okay, mitosis phase and initiation of DNA replication. During G1 phase, the cell is metabolically active. Cell is metabolically active and continuously grows but does not replicate its DNA. G1 phase corresponds to the interval between mitosis. G1, just think about your paper. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. G1 phase corresponds to interval between mitosis G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows, but does not replicate its DNA. G1 phase, the cell is metabolically active and continuously grows, but does not replicate its DNA. Okay, they synthesize protein and RNA. Okay, it's given in the notes. Most of the cell organelles duplicate. Okay, cell organelles duplicate. RNA and protein synthesize, DNA precursor, nucleotide, amino acids, sugar synthesis. Longer the phase, slower will be the division. Wait, is it? Metabolically active and continuously grows but does not replicate its DNA. Okay, during G1. फिर क्या होगा? Cell organelles. Most of the cell organelles, NELL organelles duplicates. As a synthesis phase marks the period during which DNA synthesis or DNA replication takes place. During this time, the amount of DNA per cell double. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid to n number of chromosome at Z1, even after S phase, the number of chromosome remains same, that is 2n. But number of chromatid also increases. कहाँ पर लिखो? कुछ है ही नहीं यहाँ पर. In animal cell, during S phase, DNA replications begins in the nucleus. In animal cell, during S phase, synthesis phase, okay. DNA replication begins in the nucleus. DNA replication begins in the nucleus. आई पढ़ ली बहन. In animal cell during the S phase, DNA replication begins in the nucleus, and the centriole duplicates in the cytoplasm. DNA replications begins in nucleus. DNA replication begins in nucleus. Centriole duplicates in cytoplasm. DNA replication यहीं पर लिख देते हैं ना. DNA replication और duplication in nucleus. Inside nucleus. Centriole duplication in cytoplasm. PLASM. DNA replicates inside the nucleus. Okay. DNA replication in animal cells. Okay. Nucleus and centriole duplicates. Okay. DNA replication begins in the nucleus and centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis. While cell growth continues. Okay. Centriole, tubulin protein, 9+O, cartwheel appearance. Centriole duplicates. प्रोटीन सिंथेसिस. ट्यूबलिन प्रोटीन. कौन सा वाला प्रोटीन? ट्यूबलिन प्रोटीन. ट्यूबलिन प्रोटीन synthesize tubulin protein in preparation for mitosis while cell growth continues. Special set of organelles duplicates. Chloroplast, mitochondria and Golgi bodies, RNA and protein synthesize. Okay. G नोट भी दिया है. Okay. हाई क्या ही कर सकते हैं. Prophase, metaphase, anaphase, telophase and cytokinesis. G0, G1, S and G2. G1 and S, there is one gate. Okay, checkpoint. Between S and G2, G2 and M, G2 and S, or G2 and M. G2 and M. I think G2 and M. Checkpoint. G2 and M. Okay. Anaphase, promoting complex. Spindle assembly checkpoint. Spindle assembly checkpoint. Meta to ana. Recent phase. Histone and centriole. S में ही होगा. Okay, histone protein भी यहीं पर synthesize होगा, ये बोल रहे हैं. हाँ, यहाँ दिया तो है. Histone protein synthesis S phase में ही होगा. Histone protein synthesis. क्रोमोसोम डुप्लीकेट होगा, फिर होगा तुम्हारा सेंट्रियोल डुप्लीकेशन. हिस्टोन, synthesis of histone. How do plants and animals continue to grow at their lives? Do all cells in plant divide all the time? Nah. Do you think because cells in them undergo division? Can you tell the name? Do you think all cells continue to divide in all plants and animals? No. Can you tell the name and the location of tissues having cells that divide all their life in higher plants? Okay. Apex of root and shoot, meristematic tissue. Intercalary भी होगा, वो higher plants में नहीं होगा ना, लेकिन axillary bud. Secondary growth भी तो होगा, secondary meristem भी होगा. Vascular cord cambium and vascular cambium. Do animals have similar meristematic tissues? Yes, they are called stem cells. Okay, stem cells. You have studied mitosis in onion root tip cells. It has 16 chromosomes in each cell. Can you tell how many chromosomes will the cell have at Z1? 16. Chromosomes तो increase नहीं होगा ना. after S phase and after M phase. ऑल्सो व्हाट विल बी द यहाँ पर एनाफेज में आएगा तो मुझे नहीं पता, G1 में इतना ही होगा, S में भी इतना ही होगा, M पे नहीं पता. ऑल्सो व्हाट विल बी द DNA कंटेंट ऑफ द सेल एट G1 आफ्टर S सेल एट G1 एन था तो 2N होगा G2 इफ द कंटेंट आफ्टर M फेज इज 2C 4C रहेगा. Some cells in adult animals do not appear to exhibit the division, example heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. Do not divide, cell that do not divide, example in heart. Occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit the G1 phase. These cells that do not divide further exit G1 phase to enter an inactive stage. Exit the G1 phase to enter an inactive stage called quiescent stage G0. Quiescent inactive stage, quiescent stage G0 of the cell cycle. Cells of this stage remains metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cell. Diploid somatic cell. In animals, mitotic cell division is only seen in diploid cells. Against this, the plant can show mitotic division in both haploid and diploid cells. Animals में diploid लिए मतलब mitosis देखाउँछ, whereas plant में haploid र diploid दुईटाले देखाउँछ. For your recollection of example of alteration of generation in plants, identify plant species and stages at which mitosis is seen in haploid cell. मैं completely भूल गई. zygotic mitosis हो रहा था ना एक बार, तो algae में हो रहा था. Haploid stages at which mitosis is seen in haploid. स्पोर्स से गैमेटोफाइट मैं सब कुछ क्यों भूल जाती हूँ. एक चैप्टर पढ़ा नहीं, दूसरा चैप्टर भूल जाती हूँ. देखो यहाँ पर क्या हो रहा है कि स्पोर्स गैमेटोफाइट एन है ना, गैमेटोजेनेसिस ये कहाँ नहीं एन, गैमेटोजेनेसिस फर्टिलाइजेशन हो रहा है यहाँ पर तो एकदम से. स्पोरोफाइट मियोसिस, अल्टरेशन ऑफ जनरेशन यही तो है. क्या बोल रहे हैं ये लोग. डिप्लॉइड यहाँ पर हो गया है. हैप्लॉइड कहाँ, हैप्लॉइड पे कहाँ पर माइटोसिस. हाँ तो ये माइटोसिस ही तो हो रहा है स्पोर से गैमेटोफाइट, गैमेटोफाइट से सिनगैमी तक होने गैमेटोजेनेसिस. यहाँ पर तो फिर डिप्लॉइड हो गया ना. स्पोर से गैमेटोफाइट हर जगह, स्पोर से मियोसिस गैमेटोफाइट आके स्पोर से गैमेटोफाइट जब जब आता है. हैप्लोडिप्लॉन्टिक और ये क्या है हैप्लॉन्टिक. स्पोर से गैमेटोफाइट तो माइटोसिस ही हुआ ना. ये अब तो सिलेबस पढ़ते पढ़ते एक और चैप्टर खुल गया. Plant life cycle and alteration of generation. In plants, both haploid and diploid cell can divide by mitosis. Haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies. मैं कुछ नहीं कर सकती. The ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gamete by mitosis. The plant body represents a gametophyte. Haploid plant body produces gamete by mitosis. Gametophytic generation. Okay, spores. Mitosis. This plant body represents gametophyte. Following fertilization, the zygote also divide by mitosis. आई मियोसिस लिखा है यहाँ पर, मुंह पे देखो ना, मियोसिस लिखा है डायोग्राम पे, क्या लिखा है फिर यहाँ पर? The zygote also divides by mitosis to produce a diploid sporophyte plant body. यहाँ पर लिख देती हूँ, इतना जोर जोर से ना माइटोसिस हो रहा है. माइटोसिस यहाँ पर भी माइटोसिस. The plant body represents a gametophyte. Following fertilization, the zygote also divide by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant. Haploid spores are produced by this plant body by meiosis. This in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. ग्रुप डी का क्या ग्रुप डी का? Haplontic Volvox Spirogyra Chlamydomonas cara. Volvox Spirogyra Chlamydomonas cara. Okay. Haplodiplontic Ectocarpus एक दीदी. Okay. Polysiphonia I think. Hi Priya. Can we study? अभी 19 मिनट्स बाकी है. लेट्स स्टडी ना, उसके बाद यहाँ पर ब्रेक लेंगे, तब बात करेंगे. वोल्वॉक्स स्पाइरोगायरा क्लेमाइडोमोनस. एक ई के एक्टोकार्पस केल्प्स एंड पॉलीसिफोनिया. ये हैप्लोडिप्लॉन्टिक का था. डिप्लॉन्टिक का तो फ्यूकस एंड सरगाजम फॉर डिप्लॉन्टिक फ्यूकस एल्के सरगाजम. ओके. एक्टोकार्पस पॉलीसिफोनिया एंड केल्प्स फॉर हैप्लोडिप्लॉन्टिक. एक्टोकार्पस पॉलीसिफोनिया एंड केल्प्स फॉर हैप्लोडिप्लॉन्टिक. Volvox Spirogyra and Chlamydomonas even cara. From your recollection of example of alteration of generation in plant, identify plant species and stages at which mitosis is seen in haploid cell. एम्फीज. फिर से वो एक बार एनसीईआरटी रीडिंग तो करना ही पड़ेगा, वरना मैं आज भूल जाऊंगी. यहाँ से करूँगी आज, इस बुक से थोड़ा एक बार करना पड़ेगा. वो मेरा सेकेंड टाइम होगा. ये ना मैं अभी यहाँ तक करके साइटोकाइनेसिस सिग्निफिकेंस ऑफ यहाँ तक करके मैं फिर वो पढ़ूंगी. ये तो करके ही सोंगे सोने को. तो देखते हैं क्या होता है. M phase. This is the most dramatic period of cell cycle. M phase. This is the most dramatic period of cell cycle, involving a major reorganization of virtually all components of the cell. This is the most dramatic period of cell cycle is M phase. Most dramatic period, M phase, involving major reorganization of virtually all components of cell. Major reorganization of virtually all components of the cell. Since the number of chromosome in the parent and progeny cell is same. Since the number of chromosome in parent and progeny cell is the same, it is also called as equational division. The number of chromosome in parent and progeny cell is same. चलता है. The number of chromosome in parent and progeny. Number of chromosomes in the parent and progeny cell is same. Okay, chromosome, number of chromosome same. It is also called as equational division. Equational division, M phase, equational division. The number of chromosome in parent and progeny is same. The number of chromosome in parent and progeny is same. Since the number of chromosome in parent and progeny cells is same, it is also called as equational division. Equational division. The number of chromosome in the parent and progeny cell is same. It is also called as equational division. The number of chromosome in the parent and progeny cell is the same. It is also called as equational division. Though for convenience, mitosis has been divided into 4 categories. Okay, mitosis has been divided into 4 stages of nuclear division. The number of chromosomes in the parent and progeny cell is the same. It is called as equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division. M phase. अच्छा ये पूरा M phase दिया है. मैं क्या पढ़ रही हूँ अभी? Means a reorganization of virtually all component of the cell. Since the number of chromosome in the parent and progeny cell is the same. पागल हो जाऊंगी मैं. The number of chromosome in parent and progeny is same. Number of chromosome ना, हाँ, same. It is also called equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division: prophase, metaphase, anaphase, and telophase. 4 stages of nuclear division. It is very essential to understand that cell division is progressive process and very clear-cut lines cannot be drawn between various stages. Mitosis is divided into following 4 stages. अच्छा, M phase में नहीं आता है क्या साइटोकाइनेसिस? हाँ ना, वो तो अलग ही दिया है. ये दिया है ना M phase. साइटोकाइनेसिस के बाद Z0. 4 phases है न्यूक्लियर डिवीजन. 4 न्यूक्लियर डिवीजन ऐसा कुछ. Mitosis is divided into the following 4 stages. Cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Prophase, metaphase, anaphase, and telophase. प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. हाँ, वहाँ पर जाके वो लोग ने नहीं दिया तो प्रोफेज, मेटाफेज, एनाफेज एंड टेलोफेज. Prophase, metaphase, anaphase, and telophase. Divided into 4 stages of nuclear division. It is very essential to understand that cell division is a progressive process and though for convenience, mitosis has been divided into 4 stages of nuclear division. It is essential to understand that cell division is progressive process and very clear-cut lines cannot be drawn between various stages. Prophase, metaphase, anaphase, and telophase. Okay, got it. Questions की कमी नहीं है, समय की कमी है. Prophase, which is the first stage of mitosis, follows the S and prophase, which is the first stage of mitosis. First stage of mitosis follows the S and G2 phase of interphase. Follows the S and G2 phase of interphase. In S and G2 phases, the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. Untangled during the process of chromatin condensation. The centriole, which had undergone duplication during S phase of interphase, now begins to move towards opposite pole of the cell. The completion of prophase can thus be marked by the following characteristics event. Chromosomal material condense to form compact mitotic chromosomes. Chromosomal material condense to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Initiation of assembly of mitotic spindle. The microtubules, the proteinaceous component of the cell cytoplasm, helps in the process. Cell at the end of prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, and nucleolus. And the nuclear envelope do not shows Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope at the end of prophase viewed under the microscope. Okay, colchicine is a cell poison which arrest cell division at metaphase and can induce polyploidy. Colchicine, mitotic poison, cell poison which arrest cell division at metaphase and can induce polyploidy. Okay. Initiation of assembly of mitotic spindle, the microtubule. The proteinaceous component of cell cytoplasm helps in the process. Initiation of the assembly of mitotic spindle, the microtubules. The proteinaceous component of the cell cytoplasm helps in the process. Okay. शायद नहीं हो रहा है मुझसे. चक्राते सर के साथ जीना पड़ रहा है. The continuous process of maintaining the cell. ये तो नहीं है. पूरा दिमाग आजा. 130. Centriole poleward movement. The chromosome condensation. Initial event of the condensation, chromosome condensation, appears as entangled fiber. Okay. Centriole that was duplicated in S phase of interphase starts moving towards the pole. Astral rays formation. In the initial prophase, the chromatin fiber appears like ball of wool. Spireme stays. वैसे भी ये भूल ही जाना है. Spireme stays. Two types of mitosis without astral formation: anastral or amphiastral. Centrosomes with astral rays. Mitotic apparatus, right? Late event of prophase, chromosome. नोट्स में एक चीज लिखा है. chromosomes are seen to be composed of two chromatid. Compact mitotic chromosome. Mitotic chromosome. Chromosomes are seen to be composed of two chromatid. Chromosomal material condenses to form compact the complete integration of the nuclear envelope. The complete integration of nuclear envelope marks the start of second phase. The complete disintegration of nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. Disintegration of nuclear envelope marks the start of the second phase of mitosis. Chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is mostly easily studied. At this stage, metaphase chromosome is made up of two sister chromatids. Metaphase chromosome is made up of two sister chromatids. Centromere है ना, two sister chromatid. Two sister chromatids which are held together by centromere. Small disc-shaped structure at the surface of the centromere are called kinetochore. Small disc-shaped structure at the surface of the centromere are called kinetochore. These structures serve as the site of attachment of spindle fibers. This structure serves as the site of the attachment of spindle fiber. This structure serves as the site of attachment of spindle fiber formed by the spindle fibers. To the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie रोने का दिल कर रहा है. This structure serves the site of attachment to spindle fiber. Small disc, okay. Kind of good. These structures serve as the site of attachment. This structure serves as the site of attachment of spindle fiber. Site of attachment of spindle fiber. Site of attachment of spindle fiber. Formed by the spindle fibers to the chromosome that moved into to the chromosomes that are moved into position at the center of the cell. That are moved into position. To the chromosomes that are moved into the position at the center of the cell. Hence, the metaphase is characterized. Time up. Now it's break time, okay? Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator. Hence, metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by a kinetochore to spindle fiber. The metaphase is characterized by all the chromosomes coming to lie. Hence, metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of chromosome at metaphase is referred to as metaphase plate. The key feature of metaphase are spindle fibers attached to kinetochore of chromosome. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fiber to both poles. Metaphase is characterized by all the chromosomes coming. Metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by a kinetochore. One chromatid of each chromosome connected by its kinetochore to spindle fiber from one. Leave it. मुझे नहीं सोचना है उसके बारे में. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome. One chromatid of each chromosome. कहाँ गया? Metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore at the surface of centromere called kinetochore. This structure serves as the site of attachment of spindle fiber formed by the spindle fiber to the chromosomes that are moved into position. To the chromosomes that are moved into position at the center of the cell. Metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosomes connected by its kinetochore. One chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of chromosome at metaphase is referred to as metaphase plate. The plane of alignment of chromosome at metaphase is referred to as metaphase plate. Plane of alignment of chromosome at metaphase. The key features of metaphase are spindle fiber attached to kinetochore of chromosome. Spindle fiber attached to kinetochore of chromosome. Spindle fiber attached to kinetochore of chromosomes. Spindle fiber attached to kinetochore of chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fiber to both poles. Spindle fibers attached to kinetochore of chromosome. Chromosomes are moved to spindle equator and gets aligned along metaphase plate through spindle fiber to both poles. Early prophase, late prophase. Centromere, centriole, those are centriole. हाई अब ब्रेक लेंगे। ओके, हम लोगों का ये 4th स्लॉट कंप्लीट हो गया। ब्रेक। जैसे ही ब्रेक लेती हूँ ना, कुछ खाने का दिल करता है। वैसे मेरा चार्ज नहीं है, मुझे चार्ज करना पड़ेगा, लेकिन मैं स्विच ऑफ कर दूंगी ये शायद। आर यू प्रिपेयरिंग फॉर नीट? यप नीतीश, यू आर राइट। नीतीश, भी आराम से। What kind of name is that? I don't know. लगा दो चार्ज पे, नहीं स्विच ऑफ होने देती हूँ। वैसे 2 तो होने ही वाला है ना। अगर ये नेट सही हुआ ना तो मैं एक टेस्ट दे देती हूँ, अगर नहीं हुआ तो नहीं सही हुआ तो मैं क्या ही कर सकती हूँ। कितना परसेंटेज है? आई डोंट नो, पहले ही नोटिफिकेशन आया था 20 का। 11 है। 1 आवर जितना चलेगा। मुझे तो कुछ खाने का दिल कर रहा है, लेकिन मेरे पास खाने के लिए कुछ नहीं है। किचन में जाके देखती हूँ कुछ है। फिर से एक बार राइस खा लेती हूँ ना, लेकिन नहीं खाऊंगी। मैंने अभी तक 3 बार खा लिया है, अब एक बार खाऊंगी तो 4 बार होगा और मैं मोटी हो जाऊंगी। क्या है? ये ना डार्विन में ही ग्लिच है, मैं बोल रही थी ना। हाउ मेनी डेज कुछ नहीं दिखा रहा है, बस ग्लिच ही आ रहा है। आर यू 11th 12th ड्रॉपर? बाय द वे आई एम 12th PCM। मेरा बायो था, PC भी था, एंड आई एम ड्रॉपर। मैं ना राइस खा के आई। ओके, आई लव राइस 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 राइस। हाँ अब आ गया ना, इतने मेहनत के बाद आ गया है, लेकिन मेरा प्रोफाइल नहीं आ रहा है इसमें, क्योंकि शायद ये लॉगिन नहीं होगा। मैं लॉगिन करूँ क्या? डार्विन लॉगिन, लॉगिन। फुल मॉक टेस्ट दे सकते हैं वैसे, लेकिन मैं नहीं दूंगी, मैं पार्शियल वाला ही दूंगी। प्लांट सेल द यूनिट ऑफ लाइफ, बायोमोलिक्यूल्स नहीं किया। हाँ सेल द यूनिट ऑफ लाइफ देंगे ना, उसके बाद तो सेल डिवीजन एंड सेल साइकिल देंगे। पढ़ ले, 2 बजे। कौन खाता है? नहीं मैं खाती हूँ ना, मुझे भूख लगता है। Hey, can someone tell me why are so many scholars are doing live stream of studying? Do you do it manifesting discipline? अभी खाओगे फिर नींद आएगी, पानी है ना। वैसे आज मैं इतनी बार क्यों खा रही हूँ पता है? आज क्योंकि मेरा फेवरेट है। नितिन का जवाब दे दे शुभम भाई। स्कॉलर बोल दिया, स्कॉलर, उसने हम लोगों को स्कॉलर बोल दिया। डस्टबिन चाहिए मुझे, मेरा नाक कहीं गिर ना जाए। वही तो। I was bored, that's why I started live streaming, Nitin. I think this guy, Nitin, already knows the answer, but still he is asking. I'll note it, okay? कहीं नीट के पेपर में ना आ जाए। पेन कहाँ गया? नोट कर लूँ, पेन नहीं मिल रहा, बाद में आके नोट कर लेती हूँ। ओके, I'll go for rice. I'll eat and then I'll come back. Hey Nitin, it's okay. I'm not annoyed. I was just making fun. In fact, यहाँ पर जो भी लोग हैं ना, I don't call them by their real name. Like for example, Shubham is SP. His name is Shubham, but we call there is someone else whose name is Vesh, but we call him Alex. And there is another girl whose name is Nidhi. We call her Nonu. So you are Nitin, Nitish Nitin. Well, I think the problem is they are my friends and you are not. I'll call you Nitish or something like that. Nitish. Okay. Nitish or Nitish? Nitish. Nitish ही होगा. टेस्टी है. क्या हुआ? Shubham, how many Shubhams are there? आई थिंक ये चैनल में ना सबसे ज़्यादा मैंने नाम शुभम ही देखा है. मैं तो स्पेशल हूँ, बाकी का नहीं पता. वैसे मुझे नहीं पता था कि तुम स्पेशल हो, लेकिन अभी पता चला ये प्रोफाइल देख के. तुमने बोला था कि तुम मुझे स्टॉक कर रही हो ब्ला ब्ला, और तुम्हारे पास क्या क्या खूबी है लाइक दैट. फिर मैंने बोला कि मेरे पास बहुत है कहने के लिए. फिर तुमने अपना बताया था कि तुम स्टेट लेवल बैडमिंटन प्लेयर हो, ऐसा कुछ, और तुम IIT ग्वालियर में हो. ट्रिपल IT मैडम. अच्छा अच्छा. मैंने तो इतना बड़ा के बोल दिया फिर भी. अच्छा किया डिलीट कर दिया, वरना भाई तुझे ना शुभम टाइम आउट दे देता. मत बोलिए. कुछ कुछ गंदा ही होगा, फिर मुझे बुरा लगेगा। वैसे भी मुझे आज ये 2 चैप्टर हाफ चैप्टर कंप्लीट करना है ना, अभी भी बाकी है। ब्रेक के समय ब्रेक लोगे तो सही रहेगा। ये कहना था। अरे मैं छपरी हूँ क्या? हाँ तो नहीं पता था क्या? कैप्स में लिख दूँ? चलो स्टार्ट करते हैं, अभी तो बहुत करना है ना और भी। लेट्स स्टार्ट। ओके, आई मैंने किसको पिन कर दिया? अनपिन करो। गुड लक ऑल। ये कौन सा स्लॉट है? 5th स्लॉट ना? 5 90 मिनट। शुभम, काफ़ी अजीब लगता है ना जब जो अभी कॉलेज में है वो लोग उन लोगों को एडवाइस देते हैं जिन्होंने कॉलेज कंप्लीट कर दिया है, वो भी बहुत साल पहले। पर कोई बात नहीं, होता है। इट्स ओके, इट्स ओके। आई थिंक ये लाइट ना चेंज करना पड़ेगा, यहाँ से हटा के कहीं और रख देना पड़ेगा। लाइट की वजह से लोगों को कंफ्यूजन हो रहा है कि लाइट की वजह से लोगों को कंफ्यूजन हो रहा है कि मैं अभी तक कॉलेज में नहीं गई हूँ। ओए शुभम, आर यू देयर? SRM यूनिवर्सिटी। हाँ फिर स्टार्ट करते हो कि हम दोनों? वरना मुझे भयानक भयानक एडवाइस मिलेगा। Hey, earlier I was just asking for the value of Boltzmann constant. Mistakenly I typed it wrong. Try to correct it. The send button just. चलो फिर। Okay. हाँ स्टार्ट कर दिया है सेशन। Now everyone don't text, okay? Let's study. कैसे कैसे लोग रहते हैं यार इस गोले में. वैसे मुझे लगता है कि मैं काफ़ी इमैच्योर हूँ ना, लेकिन यहाँ के लोग देखते देखते मुझे ऐसा लगता है कि मैं काफ़ी मैच्योर हो गई. स्पिंडल फाइबर अटैच्ड टू कैंटिकल ऑफ क्रोमोसोम. बच्चे तो बच्चे हैं, लेकिन बड़े भी बच्चे जैसे बिहेव करते हैं. Grow up, guys. Anaphase. At the onset of anaphase, stage. Upstage, yeah. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatid, now referred to as chromosome of the future daughter nuclei, begins to migrate towards the opposite pole. Anaphase. Each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as chromosome of future daughter nuclei, begin their migration towards the two opposite poles. Future daughter nuclei begins the migration towards the opposite pole. As each chromosomes move away from the equatorial plate, chromosomes move away from the equatorial plate, the centromere of each chromosome is towards the pole, and hence it is as each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole. And hence at the leading edge, with the arm of the chromosome trailing behind. As each chromosome moves away from the equatorial plate, the centromere of each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole. As each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole. And hence at the leading edge, with the arms of the chromosome trailing behind. The anaphase stage is characterized by The anaphase stage is characterized by the following key events: centromeres split and chromatids separate. Centromere split and chromatid separates. Chromatid move to opposite pole. Chromatid moves to opposite. हाइड कर दो रात को, लोग पागल हो जाते हैं. हाइड मतलब कि ऑफ कर दो ना चैट को. है ना, हाइड मतलब ऑफ ना. ओके कर दिया. The following key events. The following key events. At the beginning of the final stage of mitosis. Okay. The centromere of each of the equatorial plate. As each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole, and hence the leading edge with the arms of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key event: centromere split and chromatids separate. Centromere split and chromatids separate. Centromeres split and chromatids separate. Centromeres split and chromatids separate. Centromeres split and chromatids separate. Chromatids move to opposite pole. Chromatids move to opposite pole. Chromatids move to opposite pole. Centromere split and chromatids separate. Centromere split and chromosome separate. The following key events: centromeres split and chromatid separate. Chromatid move to opposite poles. Chromatids move to opposite poles. Centromere split and chromatids separate. Most chromatids move to opposite pole. The centromere of each chromosome is towards the pole, and hence the leading edge with the arm of the chromosome trailing behind, with the arm trailing behind. From the equatorial plate, the centromere of each chromosome is towards the pole. Centromere. The chromosome moves away from the equatorial plate. The centromere of each chromosome is towards the pole. Centromere. And hence at the leading edge with the arms of the chromosome. Anaphase stage is characterized by the following events: centromeres splitting and chromosome separate. Centrosome splits and chromatids separate. Chromatids move to opposite pole. Chromatid moves to opposite pole. Chromatid moves to opposite pole. Telophase. At the beginning of the final stage of mitosis. At the beginning of the final stage of mitosis. That is telophase. The chromosome that have reached their respective poles. The chromosome that have reached their respective pole decondense and lose their individuality. The individual chromosome can no longer be seen, and chromatin material, the individual chromosome can no longer be seen, and chromatin material tends to be collect in a mass in the two pole. The individual chromosomes can no longer be seen, and chromatin material tends to collect in a mass in the two pole. The individual chromosomes can no longer be seen, and chromatin material tends to collect in a mass in the two poles. Can no longer be seen, and chromatin material tends to collect in a mass. The individual chromosomes can no longer be seen, and chromatin material tends to collect in a mass. Reach their respective poles, decondense, and lose their individuality. The individual chromosome can no longer be seen. The individual chromosome can no longer be seen, and chromatin material tends to collect in a mass in two poles. The individual chromosome can no longer be seen, and chromatin material tends to collect in a mass in two poles. This is the stage which shows the following key event. Chromosomes cluster at opposite spindle pole, and their identity is lost as discrete element. Chromosome cluster at opposite spindle poles, and their identity is lost as a discrete element. Chromosome cluster at opposite spindle pole, and their identity is lost as discrete element. Chromosome cluster at opposite spindle pole, and their identity is lost as discrete elements. Nuclear envelope assembles around the chromosome. Cluster. Nucleolus, Golgi complex, and ER reforms. Nucleolus, Golgi complex, and ER reforms. Nucleolus, Golgi complex, and ER reforms. Nuclear envelope assembles around the chromosome cluster. Nuclear envelope assembles around the chromosome cluster. Nuclear envelope assembles around the chromosome cluster. Nuclear envelope assembles around the chromosome. Okay. Nucleolus, Golgi apparatus, and ER reforms. Nucleolus, Golgi complex. Nucleolus, Golgi complex, and ER reforms. Even nuclear envelopes are assembled or reappears around the chromosome cluster. Cytokinesis. Mitosis accomplishes not only the segregation of mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by a separate process called cytokinesis, at the end of which cell division is complete. In an animal cell, this is achieved by the appearance of a furrow in plasma membrane. In an animal cell, this is achieved by the appearance of furrow in plasma membrane. In animal cell, this is achieved by the appearance of furrow in plasma membrane. In animal cell, this is achieved by appearance of furrow in plasma membrane. Appearance of furrow in plasma membrane. Okay. Centripetally move. Okay, centripetally move गर्छ furrow. Appearance of furrow in plasma membrane. Appearance of furrow in plasma membrane. Animal cell. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. The furrow gradually deepens and ultimately joins the center, dividing the cell cytoplasm into plant. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Cell wall. Therefore, they undergo cytokinesis by a different mechanism. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral wall. The formation of new cell wall begins with the formation of simple precursor called the cell plate that represent, okay, centripetal in animal, centrifugal in plant, okay, that represents the middle lamella between the wall of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example, liquid endosperm in coconut. Liquid endosperm in coconut. Cell plate. Phragmoplast formed by Golgi complex to form cell plate. Okay. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral grow out to meet the existing lateral wall. The formation of new cell wall begins with the formation of simple precursor, centrifugal force. Simple precursor called the cell plate that phragmoplast formed by Golgi complex to form cell plate. Phragmoplast formed by Golgi complex to form cell plates. Phragmoplast formed by Golgi complex to form cell plate. Okay, cell plate that represents the middle lamella between the wall of two adjacent cells. At the time of cytoplasmic division, organelles like, okay, distributed. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arises. Leading to the formation of syncytium. Several nucleus in one cell. Syncytium, okay. Syncytium. Multinucleated condition arises, leading to the formation of syncytium. I 2% charge बोल रहा है. I have to charge. Syncytium. Let me charge. Charge only. Formation of syncytium, example liquid endosperm in coconut. Example liquid endosperm in coconut. Okay, we have cytokinesis. Cytokinesis, where is it? Here it is. Animal cell, I got it. Animal cell, furrow formation, construction, microfibrils, accumulated, microfilament, not microfibrils, microfilament accumulated at the central body, animal cell, furrow formation, construction, microfilament accumulated at the central body of the cell, referred as meat body. The actin and myosin filament starts pulling the wall side. Actin and myosin filament, microfilament accumulated at central body. Actin and myosin filament starts pulling the cell wall. Formation of constriction towards the center. Centripetal. Okay. Cell plate formation, plant cell. The vesicles are produced by Golgi bodies. Okay. Phragmoplast, formation of phragmoplast formed by Golgi complex to form cell plate. Phragmoplast formed by Golgi complex to form cell plate. Phragmoplast, cell plate, phragmoplast formed by Golgi complex. The vesicles are produced from Golgi bodies. The vesicles fuse together to form phragmoplast. The vesicles are produced from Golgi bodies. The vesicles fuse together to form phragmoplast. Also, the ER vesicle contributes to phragmoplast. Cell plate, plant cell. Cell wall, rigid structure, furrow or constriction is not possible. To 30. Cell plate, plant cell wall, rigid structure. Vesicle, microtubules, vesicles along with microtubules. Vesicles along with microtubules. So, cell plate, plant body, cell wall, rigid structure के ये हो गया furrow or constriction in is not possible. Vesicles along with microtubules, fuse together to form phragmoplast. Vesicles along with microtubules fuse together to form phragmoplast. Vesicles along with microtubules fuse together to form phragmoplast. The microtubules are also fused with phragmoplast. Vesicles along with microtubules fuse together to form phragmoplast. The microtubules are also fused with phragmoplast. Starts forming cell plate. The microtubules are also fused with phragmoplast. Starts forming cell plate. Precursor of cell plate is phragmoplast. Cell plate towards the periphery. Centrifugate pattern result into Ca2, calcium ion and magnesium ion. Calcium and magnesium pectate deposition. middle lamella, cell separation, okay? Mitosis. Mitosomes, uh, auxin, cytokinin, gibberellin, and auxin, cytokinin, gibberellin, and one more is there, insulin, ACGI, ACGI, CGI. In some organism, karyokinesis is not followed by cytokinesis. In some organism, karyokinesis is not followed by cytokinesis. In some organism, karyokinesis is not followed by cytokinesis. As a result of which multinucleate condition arises, leading to the formation of syncytium. Example, liquid endosperm in coconut. In some organism, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Syncytium. Example, endosperm of coconut. Liquid endosperm in coconut. Liquid endosperm in coconut. Okay. Significance of mitosis. What are the importance of mitosis? Mitosis, or the equational division, is usually restricted to the diploid cell only. However, in some lower plants and in some social insects, haploid cell also divide by mitosis. Lower plant and social organisms like bee, social insects and lower plant, haploid cell also divide by mitosis. Haploid cell also divides by mitosis. It is very essential to understand the significance of this division in life of an organism. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some example where you have studied about haploid and diploid insect? Where you have studied about haploid or diploid insect? Haploid and diploid insect. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some example where you have studied about haploid and diploid insect? Haploid or diploid insect. More mitosis usually result in the production of diploid daughter cell with it identical genetic complement, diploid daughter cell. Mitosis usually result in the production of diploid daughter cell. Mitosis usually result in the production of diploid daughter cell with identical genetic complement. Production of diploid daughter cell. Diploid daughter cell, identical genetic complement. The growth of multicellular organism is due to mitosis. Growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between of nucleus and the cytoplasm. It therefore becomes essential for cell to divide to restore the nucleocytoplasmic ratio. Nucleocytoplasmic ratio restored by mitosis. A very significant contribution of mitosis is cell repair. The cell of upper layer of epidermis, cell of the lining of gut, and blood cells are being constantly replaced. Okay, cell lining the gut, blood cells, and cells of the upper skin, cells of the upper layer of epidermis cell. Cell of the upper layer of epidermis, inner lining of gut, and blood cells are constantly replaced. Cell repair, okay. Very important contribution is mitosis is cell repair. It repairs cell. Upper layer of the epidermis, lining of the gut, and blood cells are being constantly replaced. Mitotic division is Mitotic division in the meristematic tissue, the apical and the lateral cambium. Mitotic division in the meristematic tissue, the apical and lateral cambium, vascular cambium and cord cambium, lateral cambium and apical. These are also mitotic division. Okay, the mitotic division in the meristematic tissue, the apical and the lateral cambium result in continuous growth of plant throughout their life. Okay, the significance of mitosis: cell repair, replaces old cell with new ones. The meristematic cell, lateral cambium and apical meristem also undergo mitosis for the growth of the plant. Okay, it also restores nucleocytoplasmic ratio. Cell repair. Usually produce diploid daughter cell, identical genetic complement. Growth of multicellular organism is due to mitosis. Okay, the growth of multicellular organism is due to mitosis. Cell growth result. Okay, nucleocytoplasmic ratio. Restore the nucleocytoplasmic ratio. The growth and development of organism, zygote, multicellular organization. The growth and development of organism, zygote to multicellular organization. Growth and development of organism maintains nucleocytoplasmic ratio, increase the growth of cell, helps in maintaining the proportionate component of both, maintains the surface area to volume ratio. Nucleocytoplasmic ratio. Maintains nucleocytoplasmic ratio, maintains the surface area to volume. Cell repair and replacement. Maintains chromosomal number. इतना सारा है. Maintains chromosomal number, maintaining the identity of. Helps in repair and maintenance. Lining of gut. Okay, lining of gut, blood cells and epithelial, outer layer of epithelium. Maintains chromosomal number, maintain the identity of chromosomal number, maintains the surface area volume ratio. Giant cheese egg, takoyaki. Mitotic poison. Okay, mitotic. Insulin, gibberellin, cytokinin, auxin cytokinin, gibberellin and insulin. Mitotic poison, the substance which suppresses half the division. Example, azide, cyanide, mustard gas. The substance which suppresses half the division, azide, cyanide, mustard gas. The substances which suppress or halves the division, cell division. Okay, mitotic poisoning. Colchicine checks the separation by stopping the polymerization of spindle microtubules. Checks the separation by stopping the polymerization of spindle microtubules. Allow polyploidy. Checks the separation by stopping the polymerization of spindle microtubules. Allow polyploidy. In colchicine, natural mitotic poison obtained from colchicine, liliase. Polyploidy 2x to 4x. Okay, 8 to 16. क्रोमोसोम के नंबर तो नहीं बढ़ना चाहिए ना, लेकिन यहाँ पर बढ़ता है. Colchicine poisoning. Colchicine is a cell poison which arrests cell division and at metaphase. कौन से phase में? Metaphase में. Okay, and can induce polyploidy. They induce polyploidy. Colchicine, natural mitotic poison obtained from colchicum liliase. The colchicine, natural mitotic poison obtained from colchicum liliase. Ploidy 2x to 4x. Oligopolyploidy, metaphase मान जा azide cyanide and mustard gas. Okay. Meiosis. Significance of meiosis. Meiosis 1. Okay. Generation. N equals to total time by division of one cell. N is equals to N number of time its cell undergo division. Number of time cell undergo division. N is equals to total time upon division of one cell. 2 to the power n refers to the total number of daughter cell. Total number of daughter cell 2 to the power n. 2 to the power n. Total number of daughter cell. Total number of daughter cell. N is equals to total time upon division of one cell. Total number of mitotic division is equals to x minus 1. Total number of daughter cell minus 1. Total number of daughter cell x minus 1. Total number of mitotic division. Total number of daughter cell minus 1 2 3 4 है ना 4 daughter cell minus 1 3 1 2 3 oh I see 4 minus 1 3 1 ये 1 है ना 2 and then 3 okay 1 2 3 4 5 6 7 8 8 minus 1 7 1 2 3 4 5 6 7 okay. My total number of mitotic division x minus 1. Total number of mitotic division x minus 1 to x. Number of daughter cell minus 1. Number of daughter cell minus 1. Total number of daughter cell minus 1. Okay. Meiosis referred as a reduction division. Meiosis referred as reduction division, but it has two stages: meiosis 1 and meiosis 2. Actual reductional division, meiosis 1. Actual reductional division, meiosis 1. Equational division, meiosis 2. Meiosis is the reductional division. There are two stages. Reduction referred as reduction division, meiosis, reduction division. There are two stages. The first stage is meiosis 1, actual reductional division, and the other one is meiosis 2, equational division. Equational one is reductional, the other one is equational division. Meiosis 1. One reductional division followed by one equational division. Meiosis 1 is equals to one reductional division followed by one equational division. Meiosis 1 is equals to one reductional division followed by one equational division. Reductional division, but it has two stages: meiosis 1 and meiosis 2. Equational division. Okay, meiosis 1 is equals to one reductional division followed by one equational. Meiosis 1 is equals to one reductional division followed by one equational division. Meiosis 1, one reductional division followed by one equational division. 46, 23 pair G1 S 2N S. Okay, 46 equals to 23 pair 2N S ma. Meiosis 1 23 each. Okay, S phase देखिए G1 व S phase में क्या होता है? meiosis reduction division. Daughter cell 23 each, 23 pair 23 each. 46 23 pair 23 each. Meiosis 1, cytokinesis. Daughter cell meiosis 2, reduction equation. Meiosis 23 and 23, 4 daughter cell. फर्स्ट तक कर लो 4 daughter cell. ये 4 meiosis 2 यहीं पर होगा. Meiosis 1. Lesson up. Consists of 4 subphases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, telophase, and anaphase. Prophase, metaphase, anaphase, and telophase 1. Okay. More complex, prophase, more complex, more elongated division in comparison to mitotic prophase. In comparison to mitotic prophase, meiotic prophase is more elaborate, more complex, more elongated division in comparison to mitotic prophase. Okay, more complex and more elongated division. More complex and more elongated, more complex and more elongated division in comparison to mitotic prophase. In comparison to mitotic prophase, more complex and more elongated division. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1, based on the crucial events, 5 sub-stages, phases are there. Prophase 1, based on the crucial events, 5 sub-phases are there. Sub-stages or phases are there. 5 sub-stages or sub-phases are there. Leptotin, zygotin, pakitin, diplotin, and dikinesis. Leptotin, zygotin, pakitin, diplotin, and dikinesis. Leptotin, Lady Zui. Leptotin, zygotin, please dance. Pakitin, diplotin, and dikinesis. Leptotin, zygotin, pakitin, diplotin, and dikinesis. These are the 5 subphases of prophase. 5 subphases of prophase. 1. Leptotin, zygotin, pakitin, diplotin, and dikinesis. Leptotin, zygotin, pakitin, diplotin, and dikinesis. Last is dikinesis. Okay. Leptotin, more complex, more elongated. G1, as chromatid increases, number of chromosome does not, but DNA content increases. Okay. Bouquet stays. Leptotin is bouquet stays. Leptotin is considered as bouquet stays. Chromatin condensation is observed. Chromatin condensation is observed but tangled. Chromatin condensation is observed but chromatin condensation is observed but tangled. Structure look like bouquet. Appears they arises from nuclear envelope, but tangled. Structure looks like bouquet. Appears they arises from nuclear envelope. Chromatin condensation is observed but tangled. Structure look like bouquet. Appears they arises from nuclear envelope. Leptotin. Chromatin condensation is observed but tangled. Structure look like bouquet. Bouquet stays. Chromatin condensation is observed but tangled. Structure bouquet. Appears they arises from nuclear envelope. Nuclear envelope बड़ा appear बाकस थे एक leptotin माके bouquet. Tangled. Chromatin condensation. Zygotin. The condensed chromosome starts pairing. Gene is located to 23 pair. Gene is located 23 pair at same locus. Gene is located 23 pair. 23 pair gene is located at the same locus. Synaptonymal complex material. Not material, it's maternal. Paternal or maternal. Paternal and maternal. Synaptonymal complex. The condensed chromosome starts pairing. Condensed chromosome starts pairing Pairing is done between homologous chromosome. Pairing is done between homologous chromosome. Pairing is done between homologous chromosome. Pairing is done with the help of protein complex or synaptonymal complex. Two homologous chromosome. Two homologous chromosome. Synaptonymal complex. Synaptonymal complex. Pairing is done with the help of complex protein or synaptonymal protein, 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 complex. Protein complex or synaptonymal is done with the help of protein complex or synaptonymal. Synaptonymal complex protein is done with the help of protein complex or synaptonymal complex. Protein complex or synaptonymal complex. The condensed chromosome starts pairing. The condensed chromosome starts pairing. Pairing is done between two homologous chromosomes. Pairing is done between two homologous chromosomes. This is zygote, okay, a part of prophase. The condensed chromosome starts pairing. The condensed chromosome starts pairing. Pairing is done between two homologous chromosomes. Pairing is done between two homologous chromosomes. Gene is located to 23 pair at the same locus. Synaptonymal, maternal and paternal synaptonymal complex. Pairing is done with the help of two homologous chromosome. What kind of chromosome? Homologous. Two homologous chromosome. A synaptonymal complex. Synaptonymal complex. Pairing is done with the help of protein complex or synaptonymal complex formed between non-sister chromatid of homologous chromosome. Formed between नॉन सिस्टर क्रोमैटिड ऑफ होमोलोगस क्रोमोसोम. आया ये होमोलोगस का क्या मतलब है? बेटा I'll take a break. I'll take a break, okay? Plant cannot any structural organization is still the unit of life. Which of the following is responsible for the origin of lysosomes? Chloroplast, mitochondria, Golgi bodies, ribosomes. I think it's Golgi body. Which of the following organelle is located near the nucleus and contains a collection of flattened membranous bound cisternae? I think it's Golgi apparatus. Cytoskeleton is made up of cytoskeleton, okay, cellulosic microfibrils, cellulose deposition, proteinaceous filament, calcium carbonate granules. Cytoskeleton is made up of cellulosic microfibrils. I may be wrong, okay, in this one. Cytoskeleton, right? Proteinaceous structure present. Okay, an elaborate network of filamentous proteinaceous structure. Resume. प्रोटीनेशियस फिलामेंट ना अभी एनसीईआरटी था मैं भूल गई कहाँ पर गया पेन आई नीड पेन फिलामेंटस प्रोटीनेशियस यहाँ पर तो एनसीईआरटी का लाइन ही था ये तो cytoskeleton an elaborate network of filamentous proteinaceous structure okay ये है शायद cytoskeleton filamentous proteinaceous structure प्रोटीन का और फिलामेंट का proteinaceous filament. Ribosome RNA is actively synthesized in nucleolus. Mitotic spindle is mainly composed of mitotic dash protein. Spindle, myosin actin. Mitotic spindle. Centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by amorphous pericentriolar material. Both the centriole in a centrosome lie perpendicular to each other, in which each has an organization like cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplet are also linked. The central part of proximal region of centriole is also proteinaceous and called the hub, which is connected with the tubules of peripheral triplet by radial spokes made of protein. The centrioles form the basal body of cilia and flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cell. Microtubules. Resume. Mitotic spindle is mainly कहाँ पर है ये वाला? The centriole form the basal body of cilia and flagella. Centriole, spindle fiber, spindle apparatus. I don't know. Mitotic spindle is mainly composed of protein, tubulin protein. The best material for the study of structure of cell membrane is muscle cell, liver cell. The best material of the study of structure of cell membrane. Privacy of human. Non-membranous nucleoplasmic structure in the nucleus are the site for the active synthesis of rRNA. Mark the mismatched amyloplast Allyloplast, chloroplast. The type of ribosomes found in prokaryote cell, 70S. Plant cells differ from animal cells in having cell wall, plastid, and a large vacuole. All of this. Mitochondria and chloroplast are semi-autonomous organelles. They are formed by division of pre-existing semi-autonomous organelles. They are formed by division of pre-existing organelles as well as contain DNA but lack protein synthesizing machinery. Which of the following functions are function of lysosome? All of the above. Initiation of mitosis, aid in fertilization, autolysis. इतना सब कुछ आता है क्या? The tip of chromosome are known as telomere. Which of the following organism is not an example of eukaryotic cell? E. coli, Euglena viridis, amoeba. Which of the following organism is not an example of eukaryotic cell? Paramecium, Escherichia coli, Euglena, amoeba, paramecium. पक्का E. coli होगा ये, बैक्टीरिया होगा ना. Which of the following statement are correct? Mitochondria contain DNA, correct. 70S ribosomes occurs in prokaryote, correct. Ribosomes are made up of phospholipids and oligosaccharides. Ribosomes are not found in Protista and Monera. 4 is incorrect. 1, 2, 3. Ribosomes are made up of phospholipid and oligosaccharide. Granular structure, ये इसमें मतलब प्रोटीन और वो होगा ना, RNA. Ribosomes are the granular structure first observed in electron microscope as a dense particle by George Palade. They are composed of ribonucleic acid, RNA, and protein, and are not surrounded by any membrane. 1 and 2 ही है. They are not surrounded by they are prokaryotic ribosome, 70S. Okay. They are not surrounded by any membrane. RNA, ribonucleic acid and protein. They are composed of ribonucleic acid and RNA and protein. RNA and protein. The factors which influence membrane fluidity are cholesterol, carbohydrate, percentage of unsaturated fatty acid receptors. Cholesterol, carbohydrate, percentage of unsaturated fatty acid. 1 and 2. Receptors, cholesterol. The concept of omnicellular, acellular. Rudolf Virchow. Respiratory enzymes are present in the following organelles: peroxisomes. Prokaryotic cells are characterized by absence of chromatin material, absence of nuclear membrane. The nuclear envelope acts as an interface between the genetic component of the cell and the cytoplasm. It thus protects DNA against the mutagenic effect of cytoplasmic enzyme. The content of inner compartment of mitochondria is called matrix. The outer membrane forms number of unfolding. Okay, so but the reason is false. Smooth endoplasmic reticulum. Glycosylation, lipid synthesis. ACO2. B. Rough endoplasmic reticulum. Protein synthesis. BCO1. Golgi complex. CO3. Spindle formation. Peroxisomes have ribosomes. Peroxisomes. Microtubules, or they are not. They are peroxisomes. Various enzymes. They have various enzymes. Ribosomes, DNase, catalase enzyme. Catalase enzyme. Peroxisomes have catalase enzyme and Peroxisms have catalase and peroxidase enzyme. Catalase and peroxidase enzyme. Pili in bacteria represents cilia. Extra chromosomal genetic outgrowth of cell membrane on donor. Cilia, flagella. Extra chromosomal genetic outgrowth of cell membrane of donor cell. Aya, pili. RER, intracellular protein synthesis and secretion. ACO3. BCO2. Which of the following structure is the lysosome? Which of the following structure between two adjacent cells is an effective transport pathway? Plasmodesmata. Plastoquinone. Between two adjacent cells is an effective transport pathway. Plasmodesmata, okay. The organelles that are included in endomembrane system are endoplasmic reticulum, mitochondria, endoplasmic Golgi, lysosomes, and vacuole. Find out the incorrect statement from the following. Enzymes packed in lysosomes are made through rough chloroplast. Rough endoplasmic and produce proteins and lipids respectively. Lysosome is related with the destruction of production. Shut up. Lysosome is related with the destruction of products which enters through plasma membrane. Nucleic acid is present inside the nucleoplasma of eukaryotic nucleus. Destruction of product which enters through plasma membrane. फर्स्ट वाला ही गलत होगा. Enzymes packed in lysosomes are made through rough chloroplast. क्लोरोप्लास्ट से थोड़ी बनता है. एनिमल प्लांट दोनों में है वो. लाइसोसोम की बात हो रही है. If the nucleus is a cell controlled entered and chloroplast its solar collector. If the nucleus in a cell controls center and chloroplast its solar collector. Which of the following might be called the cell combination food processor and garbage dump? क्या है? किस बकवास घटिया क्वेश्चन दिया है. Contractile vacuole is present in the endoplasm of amoeba. They are responsible for osmoregulation. Contractile, 1 and 2 are correct. Endoplasm of amoeba. If the nucleus is a cell control center and chloroplast control center and chloroplast its solar collector. Chloroplast solar collector, nucleus cell control center. Which of the following might be called the cells combination food processor and garbage disposer? Golgi apparatus. ये 20 का क्यों नहीं किया? The nuclear envelope act as interface between the genetic component of the cell and the cytoplasm. एक छोड़ा है, 3 गलत और 26 राइट. 3 wrong and 1 skip. Okay. Cytoskeleton is made up of proteinaceous filament. Mitotic spindle is mainly composed of mitotic spindle is mainly composed of Protein. Tubulin protein. Mitotic tubulin is mainly composed of tubulin protein. Mitotic spindle. Mitotic spindle composed of protein. The best material for the study of structure of cell membrane. RBC of human. Best material for the study of structure of cell membrane is, ये वाला गलत हो गया ना, autolysis. All of the above में autolysis था. Which of the following is the function of lysosome? Autolysis. अब ये autolysis का क्या मतलब होता है? Autolysis. Autolysis, correct. Indeed involved in autolysis, which refers to the process of self-lysation within a cell. Autolysis. Lysis. Autolysis function. Self-lysation. Function of lysosome, autolysis. Autolysis, okay. 16 का क्या था? 1 and 3. The factors which influence membrane. The factors which influence membrane fluidity are cholesterol and percentage of unsaturated fatty acid. Okay. Cholesterol and percentage of fluid mosaic, quasi-fluid nature enables lateral movement of protein. Fluidity. Cholesterol and the factor influence. Cholesterol. Unsaturated fatty acid. Fatty acid. Affects. Percentage of unsaturated fatty acid. Percentage of unsaturated fatty acid, which influence the membrane fluidity. Respiratory enzymes are present in the following organelles. Mitochondria. Respiratory enzymes are present. Respiratory enzymes are present. Aerobic respiration. Specific two members have their own specific enzymes associated with mitochondria. Respiratory enzyme present. Extract E from carbs to form ATP. Energy from carbs to form ATP. Okay. Enzymes. Respiratory enzymes. Respiratory enzymes. If the nucleus is a cell control center and chloroplast its solar collector, might be called as cells combination food processor and garbage disposal. Lysosome. Garbage disposal. Lysosomes. Hey, I'll sleep now. I'm kinda sleepy. I haven't completed it, but I'll sleep. I gave the test. If you're there, bye Shubham. If you want to study, you can, but I'll sleep, okay? Bye all. Now you can text. You'll study or you'll also sleep. My journey, India to the UK. Avantika Guru. Oh, okay, तभी मैं सोचूं. My God. हाँ अभी एक दो क्वेश्चंस करके. ओके. गुड नाइट. गुड लक तुम्हारे लिए. मैं तो सो जाऊंगी अब.